going to begin today. And today we have the presentations of significant educational achievements by the professors of the teaching disciplines. And these significant educational achievements for the year 2020 presented by the professors in the pre-recorded mode of the teaching disciplines of this institute. And this is a very important activity that we carry out during our convocation program. And this will be presented in the school mode. And Dr. Seema Jaggi, Professor, Discipline of Agricultural Statistics, ISRI, is the convener of these presentations. So in the forenoon sessions, in the first session, which is we have School of Crop Improvement and School of Crop Protection and School of NRM. So with this brief introduction, I would like to welcome all our resource persons, the chairman of the different sessions. First, we would like to heartily welcome Dr. Shalja Hitalmani, who will be chairing the first our session on the School of Crop Improvement. So in this we have 3 presentations to be made by the professors, and they are from genetics and plant breeding, and then we have plant genetic resources and seed science and technology. So the total presentations would cover 37 theses. And in this, there are 22 MSc presentation students presentation, and 15 PhD students will be covered. So let me share here that this is the partial educational and research achievements carried out by 37 students under School of Crop Improvement. So the chairman of today's first session, Dr. Shailaja Hitalmani. She was former Dean, Postgraduate Studies and Emeritus Professor ICR in UAS Bengaluru. She has played a crucial role in development of aerobic rice and also received the prestigious Rao Bahadur Dr. B. Vishwanath Award of IRI in 2013 for her contributions to agricultural research. So first of all, on behalf of Director IRI, faculty, and on my own behalf as Dean IRI, I welcome Dr. Shailaja Hitalmani from the core of my heart. It's my pleasure to welcome our Honorable Director, Dr. A.K. Singh, who had been very dynamic and guiding us all through. And I heartily welcome all distinguished dignitaries, invitees. I would like to welcome Dr. H.B. Singh, who has already joined, who is the chairperson in the next session, and all the joint directors, project director, heads of the division, professors, faculty, staff, students, ladies and gentlemen. So all of you are cordially invited on the 3rd day of IRA Convocation celebrations. Now I would like to invite Dr. A.K. Singh, our director, to introduce chairperson to all the invitees and the attendees. Thank you.
Thank you, Dr. Asmi, and very good morning to each one of you. I would like to extend a warm welcome to the professors' presentations that we are going to have today. And to begin with, we have the School of Crop Improvement, where we have the professors from different disciplines. They will be making presentation. And to chair the session, we have Dr. Shailaja Hitalmani, an eminent geneticist and plant breeder. I can also see Professor R.M. Singh, my brother from Banaras Hind University, who retired as Dean Faculty of Agriculture. He has also joined. Thank you, Aisha, for being here. And it's a pleasure to see you here. And Dr. H.B. Singh, who has been a grand senior of mine in Banaras Hind University, who is going to chair another session on plant protection. All the professors, heads of the divisions, students, and friends. We have about 304 participants with us who have joined us as attendee in the program. All the students and faculty members, I extend a warm welcome to each one of you. To tell about Dr. Shailaja, I have known her for very long and as a role model in the field of genetics and plant breeding. She did her graduation, post-graduation from US University of Agriculture Sciences, Bangalore, also PhD from there, and then did postdoc at International Rice Research Institute. And she is one of very few pioneers who established the DNA marker-assisted lab early in 90s, when people had not, in fact, started talking about DNA markers. Established as US University of Agriculture Sciences, Bangalore, the first lab of marker-assisted breeding, and made pioneering contribution to development of varieties of finger millet and rice, particularly suited for heroic condition using molecular breeding. She is decorated with many prestigious awards, like ICR Young Women's Agriculture Scientist Award, Dr. C.V. Raman Research Award, Hariom Ashram Trust Award, Rao Bahadur B. Viswanath Award, very prestigious award of IRI that was conferred upon her some 2 years back, if I remember correctly. Salja is also fellow of National Academy of Agricultural Sciences. And she has been an involved teacher as professor and head department of genetics and plant breeding and dean of the Faculty of Agriculture at US Bangalore. Her contribution is monumental, and it's our great pleasure, Dr. Salja, to welcome you for this program. And we look forward to your remarks at the end of presentations made by the professor. Once again, welcome to each one of you. Thank you very much.
Thank you, Dr. Singh. I think we can start.
Thank you, sir. Yes, yes, ma'am. I would like to invite Dr. Seema Jaggi to take up the proceedings further. But before Dr. Seema, we would like to have Dr. Shailja Hitalmani's initial remarks, ma'am, please.
Okay. Thank you. Thank you, Rashmi. And also I thank Dr. A.K. Singh, director and my longtime colleague, to have invited for this very interesting program, because involving with student has been very close to my heart, and I have done that for last 35 years. I have been a teacher basically and then researcher, and we have been involved with several postgraduate research which are useful to the farmers actually. And I saw the list that you sent to me also. Most of them have application oriented research by your prestigious teachers at IRI. So I'm very happy to see some of them, and I'll be happy to hear the results also now. And I thank all the persons involved here for inviting me, and I congratulate the graduating students and the awardees at this program. And since we are already late because of me, I wish all of them the best wishes. And shall we start the program?
Yes, ma'am. Yes. Thank you so much for your kind words. And now to Dr. Seema Jaggi to take up the further proceedings.
Thank you, ma'am. Good morning to all of you, and welcome to this special session on significant research achievements of postgraduate students for the year 2020-21. Today is the third day, as ma'am told, of the 59th Convocation program. So as per the tradition, today and tomorrow, the professors of all the 26 disciplines will be making presentations of the educational achievements of the students of their respective disciplines. This year, a total of 248 students will be receiving the degrees, including 101 PhD students. The presentations will be made in school-wise mode. Again, because of this pandemic situation, it was decided to have these presentations in virtual mode, and further also to have it in pre-recorded format. So this was really a great experience for all of us to learn this mode of presentation, and we all really enjoyed this. All the professors were allocated the time for their presentation, which was proportional to the number of students receiving degree in each discipline. So we'll be playing the recorded presentations of these professors. To begin with, we have 3 presentations in this session of School of Crop Improvement. So first presentation is from the discipline of genetics and plant breeding, and Professor Vinod is making this presentation. So I will request Dr. Mishra.
Before you start, Dr. Mishra, I would like to add that, yesterday and day before yesterday, we had the presentation for gold medal by the selected students from different disciplines. So MSc and PhD students from all 25, 26 disciplines identified one from each discipline. They presented their work yesterday and day before yesterday before a 5 members jury. And today we will be announcing the results of those gold medal winners also. But in addition to that, the other remaining students whose work could not be presented by them, we have a tradition of presenting the significant research finding of those students by the professor of respective discipline. And since this was happening in online mode, we decided in order to economize time to have the presentation pre-recorded. All the professors are here present in this meeting, and after the presentation is over, if there is any query, questions that can be addressed by the respective professors, and before we take the remarks from the chairperson. So thank you very much, and now you can go ahead with the program.
Yes. Dr. Mishra, please.
To all. Respected Chairman, Director IRI, Dean and Joint Director Education, Professors, Heads of Divisions and students. Here I am making a brief presentation of significant research achievements of postgraduate students. The mandate of the division includes basic and strategic research, applied research, transfer of technology, human resource development, and developing and strengthening collaborative linkages with national and international organizations. The research program of our students are oriented to meet the mandate of the division. The division of genetics, we are having 61 faculty members, out of which 52 are located in Delhi and 9 are from regional stations. Total 21 students are getting their degrees, 12 MSc students and 9 PhD students. In the areas of research, the major areas covered are biotic stress tolerance, abiotic stress tolerance, nutritional quality, hybrid breeding, yield and other economic traits, and nutrient use efficiency. The crops covered are wheat, rice, maize, lentil, chickpea, pearl millet, and mustard. 21 students have contributed to this presentation. 12 are MSc students and 9 are PhD students. In the area of biotic stress tolerance, 5 students have undertaken research work. The areas addressed are leaf rust in wheat and foliar blast in pearl millet. PhD student Ms. Kirti Rani worked on genetics and molecular mapping of leaf rust resistance in agilotomography-derived introgression lines. The major objectives were to identify cytologically stable plants and mapping of rust resistance gene. Introgression line ER9-700 was found to be cytologically stable, and genetic analysis revealed a single dominant gene for leaf rust resistance. Mapping of leaf rust resistance gene in ER9-700 was done using both SSR and SNP markers. The leaf rust resistance gene, tentatively named as LRM, was mapped on chromosome arm 2AS, and 3 SNP-based PCR markers were the closest to the gene at a distance of 2 centimorgan. The work identified a novel leaf rust resistance gene, and the work was published in the journal Theoretical and Applied Genetics. Another student, Mr. Sekha Chaudhary, worked on characterization of Triticum militini derived introgression lines for leaf and stipe rust resistance. 45 Triticum militini introgression lines were evaluated for leaf and stipe rust resistance. 14 introgression lines were identified to carry leaf rust resistance at seedling stage, while 9 lines were found to be resistant to stipe rust. Molecular characterization revealed maximum introgression in A genome and minimum in D genome. The introgression line TMD11-3 showed maximum introgression with 28.1% alleles from Triticum militini, while TMD2-7 was found with minimum introgression. Analysis of selected TMD lines was undertaken. TMD11-6 was found to carry a single recessive gene for leaf rust resistance, while 2 recessive genes were found to control resistance in TMD13-4. MSc student Mr. Deepak worked on genome-wide analysis of nitrate reductase and S-nitrosoglutathione reductase family genes in response to leaf rust infection in wheat. Genome-wide analysis has identified 9 nitrate reductase genes and 3 nitrosoglutathione reductase genes present in bred wheat. Expression analysis at different time interval after inoculation with leaf rust pathogen has observed upregulation of NIA1, 2, and 3 at 24 hours after inoculation, while GSNOR2 was found to be highly upregulated at 24 hours after inoculation, which indicates that their involvement in the process of leaf rust resistance. MSc student Mr. Ramesh worked on genome-wide analysis of thyrodoxine family genes in response to leaf rust infection in wheat. Genome-wide analysis has identified 42 thyrodoxine genes present in bread wheat genome. The expression analysis of selected thyrodoxine genes at different time interval after inoculation with leaf rust pathogen revealed the upregulation of TRX2-1A, TRX6-1D, TRX11-5A, which indicates their involvement in leaf rust resistance. PhD student Mr. M. Malik worked under guidance of Dr. S. P. Singh on genetics and molecular mapping of blast resistance in pearl millet. Genetic analysis showed a single dominant gene for foliar blast resistance in genotypes PPMI 10018 and PPMI 2464. Test of alleles determined that both the lines carried the same resistance gene. Molecular mapping of blast resistance gene in genotypes PPMI 10018 and PPMI 2464 was undertaken, and the resistance gene was found to be located on chromosome 4. IPES 0076 was the closest marker linked to the resistance gene. In the area of abiotic stress tolerance, drought and heat tolerance were the major traits studied by the students. 2 PhD students and 1 MSc student worked on abiotic stress tolerance. Mr. Harisha, MSc student worked on phenotypic and molecular characterization of wheat genotypes for glaucousness under drought and heat stress. The objectives were to assess the role of glaucousness in imparting tolerance to drought and heat stress and to study the polymorphism in the genes QTLs associated with glaucousness. Total 100 genotypes were selected, of which 30 were non-glaucous and 30 glaucous reels from the cross SD29C2 into a synthetic 46, and remaining genotypes included some mutants and other gen plus lines. Under irrigated conditions, maximum number of genotypes expressed non-glaucous phenotype, whereas under RI and LS conditions, there were many genotypes that expressed highly glaucous phenotype as compared to irrigated conditions. Putative SNPs associated with glaucousness in reels were identified on chromosomes 2B and 2D. Mr. Rahul Gajgate, PhD student, worked on QTL mapping for drought tolerance in bread wheat. The objectives were characterization of reels for drought tolerance, related traits under different moisture regime, and QTL mapping of reel population derived from the synthetic 46 into SD29C2 for drought tolerance and related traits. For genotyping, 35K SNP chip was used, a linkage map was constructed, and QTL mapping was undertaken. In this study, 16 major QTLs with more than 10% phenotypic variance were identified for different traits. QTL hotspots for component traits of drought tolerance were identified on chromosome 4B, 5B, and 7A. Mr. O.P. Patidar worked on genetic and molecular characterization of terminal high temperature stress tolerance in Brassica gentiana. The major findings of the study are that correlation and path analysis showed that under late zone condition, plant height, number of secondary branches, and number of siliqua on main shoot can be used for indirect selection. Di-allele analysis of genotypes revealed the presence of significant variability among genotypes for combining ability. Genotypes PM26, Bio902, and NPJ119 were shown to have better GCA than others. One MSc and one PhD student worked in the area of heterosis breeding. Mr. Tribhuvan, PhD student, worked on inheritance, molecular mapping, and validation of fertility restorer genes for A5 cytoplasm in pearl millet. The major finding was that the fertility restoration for A4 cytoplasm is governed by a single dominant gene. Of the 30 hybrids tested, 16 had reported positive grain yield heterosis over PUSA 1201. Linkage map was constructed and fertility restorer gene RF5 was located on chromosome 2. RF5 was mapped 5.7 centimorgan distal to IPES 007, the closest marker. MSc student worked on development and understanding of heterotic groups in subtropical maize. The objectives were morphological and molecular characterization of elite maize inbred lines and grouping of maize inbred lines based on their heterotic pattern. The total number of genotypes used in the study was 30. Morphological trait-based grouping of inbred lines had clearly classified 30 genotypes into 3 clusters. On combining ability, 30 inbred lines were classified in 3 heterotic groups. 8 inbred lines were grouped in heterotic group A, 11 lines were grouped in heterotic group B, whereas 11 of them were grouped to separate heterotic group AB. 5 MSc students worked on yield-related traits and some other morphological traits. Mr. Amaresh, MSc student, worked on genetic and molecular analysis of seedling stage zebra leaf mutation in rice. The seedling stage zebra leaf was found to be a monogenic recessive trait. QTL-seq analysis revealed a distinct peak in chromosome 2 delimited to 0.25 MB on chromosome 2. Till date, no gene governing zebra leaf mutation has been mapped from chromosome 2. Therefore, the mutation is a novel mutation and the gene governing seedling stage zebra mutation in PUSA zebra 18 seems to be a novel gene. Ms. Harshita, MSc student, worked on genetic analysis of uppermost internode in rice. The objectives were morphological and molecular characterization of rice germplasm for uppermost internode, genetic analysis of uppermost internode in rice, and mapping gene using bulk segregant analysis. The inheritance study and the segregation pattern of elongated uppermost internode in F2 confirms that the trait is controlled by single gene. Based on composite interval mapping, a gene was mapped on chromosome 6 for uppermost internode with a load value of 57 and I-square value of 0.50. Mr. Dana Kumara, MSc student, worked on association mapping for seedling root architecture in diverse set of wheat germplasm. The objectives were to study the genetic variation of seedling root architecture and root angle in diverse panel of wheat and to identify marker trait associations with seedling root architecture traits through genome-wide association analysis using 35K SNP arrays. Salient findings of this study are given here. Population structure analysis stratified the association panel into 2 structured subpopulations, and candidate genes such as formin homology 1, ubiquitin-like domain superfamily, ATP-dependent 6 phosphofructokinase were identified from the associated genomic regions. MSc student Amish Asuni worked on characterization of recombinant inbred line population for pod and seed traits and validation of markers linked to flowering time genes in chickpea. Characterization of real population in chickpea for flowering time, pod and seed traits was undertaken. Transgressive segregation was observed for pods per plant, seeds per plant, seed size and seed yield except time of flowering. The study has shown that it was possible to combine early flowering and maturity with relatively high seed yield potential in chickpea. Molecular marker analysis showed that amplification pattern of EFL1 gene-related marker TR13 of chickpea was similar to that of marker CAELF3A-F1 linked to ELF3 flowering time gene in Arabidopsis, suggesting the presence of some conserved genomic regions controlling flowering time in the 2 species. Ms. Ambika, MSc student, worked on genetic analysis of yield traits and identification of markers for stem growth habit using bulk segregant analysis in chickpea. The major findings are inheritance study showed that semi-determinate growth habit of BG3078-1 is governed by a single dominant gene DT2, and its genotype was designated as recessive homozygous DT1 and dominant homozygous DT2. 3 SSR markers were identified as putatively linked to DT1 locus, and considerable variability was observed for all the traits studied, and transgressive segregants were also identified. In the area of quality improvement, 3 PhD and 1 MS student worked. Mr. P.C. Gyani, PhD student, worked under the guidance of Dr. A.K. Singh on genetic analysis and molecular mapping of low phytic acid trait in rice. The objectives were to understand the genetic basis and inheritance of the low phytic acid trait in the LPA mutant, POSA low phytic acid mutant 11, PLM11, molecular mapping of low phytic acid trait in PLM11, and marker state integration of LPA trait in the genetic background of popular basmati rice variety PB1121. Major findings of the study are: genetic analysis of LPA in PLM11 indicated that the trait is quantitative in nature. Molecular mapping of LPA trait in PLM11 identified a major QTL QLPA 8.1 explaining 22.2% of the PVE on chromosome 8, and the PCR-based marker RM25 and RM22832 were identified flanking the QTL. These markers were used in marker state selection. The in silico analysis was suggestive of a locus encoding for a protein similar to phosphatidyl inositol 4 kinase family member with a possible activity in the lipid-dependent biosynthesis of phytic acid. PhD student Ms. Bhuvneshwari also worked under guidance of Dr. A.K. Singh on characterization of Manipur aromatic rice germplasm and genetic improvement of Manipur black rice cultivar Chaho Poireton. Molecular analysis revealed 3 subpopulations present in aromatic rice germplasm from Manipur. Total anthocyanin content estimated through LCMS analysis revealed that anthocyanin content varied in a range of 29.8 to 275.8 mg per 100 gram dry weight. Major component in anthocyanin pigments was found to be cyanidin 3-O-glucoside. The findings have already been published in the journal Frontiers in Genetics. QTL-seq analysis of 2 extreme bulks identified a genomic region from 27 to 28 MB of chromosome 4 responsible for black pericarp color pigmentation in Chaho Poireton. To improve Chaho Poireton, novel dwarfing allele from semi-dwarf rice cultivar PUSA 1652 was used as a donor, and the novel ST1 allele present in the PUSA 1652 was characterized and a DCAPS marker was developed. The findings are already published in the journal Plants. Black rice landrace Chaho Poireton was improved by introducing novel dwarfing allele ST1, and dwarf nils were developed using foreground and background analysis. Mr. Yatish, PhD student, worked on integration of low phytate trait into elite inbred lines of maize through marker state backcross breeding. The objectives were to integrate genes determining low phytate trait into parental lines of maize hybrids and to understand the effects of LPA1 and LPA2 on phytic acid content. Parental line of hybrid DHM121 were used as recurrent parent. Foreground and background analysis was done to develop near isogenic lines of the parental lines. The improved nils were developed for BML6 and BML45 having phytic acid content of 1.67 and 1.80 mg per gram compared to recurrent parent having phytic acid content of more than 3 mg per gram. MS student Mr. Sridhar Ragi worked on morphological and molecular characterization of mass-derived LPA1 and LPA2 based low phytic acid genotypes in maize. The objectives were to evaluate the LPA1 and LPA2 based maize inbreds for agronomic performance and yield related traits, and to analyze the genetic diversity among LPA1 and LPA2 based low phytic acid maize inbreds using microsatellite markers. A total of 24 LPA1 and 24 LPA2 based inbreds along with 7 wild type maize inbreds were used in this study. 36.2% reduction of phytic acid with increase in inorganic phosphorus was observed in LPA1 based inbreds, while in LPA2 based inbreds 24.3% reduction of phytic acid with increase in inorganic phosphorus was observed. SSR-based molecular diversity analysis grouped LPA1 and LPA2 inbreds in 3 clusters. The information can be further used in hybrid breeding. Another important area in which our students worked is nutrient use efficiency. One MSc and one PhD student worked in this area. MSc student Miss Amrita Thomas worked on genetic and molecular studies for elucidating phosphorus response in PUSA 44 PUP1 near isogenic lines. The objectives of the study were to assess the performance and adaptation of the PUSA 44 PUP1 nils under multiple environments having varying phosphorus status, to characterize the root associated traits in PUSA 44 PUP1 nils, and to characterize the transcriptome level variation in PUSA 44 and its PUP1 nil. The major findings of the study are: PUSA 44 PUP1 nils showed significant yield advantage ranging from 11.4% to 194% over PUSA 44 when grown under low phosphorus conditions. PUP1 QTL improves all root system architectural traits except for root diameter, which reduced under low phosphorus conditions. Without PUP1, low phosphorus induced significant stress response in PUSA 44, leading expression of genes to program cell death, stress defense, and DNA repair. And addition of PUP1 QTL provided leverage to PUSA 44 by enhancing the expression of genes related to growth, nutrient acquisition, and phosphate metabolism. PhD student Mr. V.R. Prakash Reddy worked on association mapping for key traits influencing phosphorus uptake and utilization efficiency in mung bean. The major objectives were characterization of diverse mung bean genotypes for traits influencing phosphorus uptake and utilization efficiency, molecular characterization of diverse mung bean genotypes, and identification of molecular markers linked to the traits influencing phosphorus uptake and utilization efficiency by using association mapping approach. 195 diverse genotypes of mung bean were characterized for phosphorus use efficiency. The genotype PUSA 1333 showed best performance for total root length, total surface area, total root volume, root dry weight, shoot dry weight, and total dry weight under both optimum phosphorus and low phosphorus conditions. Traits like total root length, total shoot area, total root volume, root dry weight, shoot dry weight, and total dry weight are sufficient to explain the most of the variation and might be used for screening of phosphorus use efficiency in mung bean. These findings have been published in journal PLOS ONE. NGS-based GBS was performed in 144 diverse genotypes of mung bean, and a total of 55,634 SNPs were anchored across all the chromosomes of mung bean. Marker trait association analysis was performed, and total 231 SNPs were found to be present in 122 genes. 173 SNPs were present in 92 protein coding genes, and remaining 58 SNPs were present in 26 uncharacterized genes. Arabidopsis orthologs of 18 genes were associated with nutrient uptake and root architectural development pathways, and these are putative candidate genes. 3 genes were identified with missense SNPs in their CDS regions, and these are potential candidate genes. The findings have been published in Frontiers in Plant Science. Our faculty members received various awards and honors, including NAS Fellowship. This is another honor for our faculty. Our faculty member Dr. A.K. Singh, who is also Director IRI, appeared in the program Life in Science with Pallav Bagla, discussing about the story of Basmati rice. Several of our students received various awards. Mr. Amit Kumar and Mr. Vidyasagar were recipients of Jawaharlal Nehru Award for Outstanding Doctoral Thesis Research in Agricultural and Allied Sciences. Similarly, Abhijit Kumar Das received IRI Merit Medal. The work of our students are published in several prestigious journals such as theoretical and applied genetics, frontiers in plant science, frontiers in genetics, plants, PLOS ONE, and many other journals of international repute. All our students are well placed. Many of them joined ARS and similar positions, and others are continuing their PhD at IRI. With this, I end my presentation, and I thank each and everyone for patient hearing.
Thank you, Dr. Vinod, for nicely compiling and presenting the work done by 21 students. I think we'll have the discussion after all the three presentations are over, if the chairman permits. Okay. So now I would like the presentation by Professor Veena Gupta, Plant Genetic Resources. She'll be presenting the work done by 8 students.
Good morning, everyone. Respected chairman of the session, other dignitaries present, and dear students. I'll be briefing you about the salient achievements made by the students in the PGR discipline. During the year, we have 4 MSc students and 4 PhD students who have conducted their research work. So 3 of the MSc students have worked on cowpea germplasm, and the first student is Mr. Siddhant Ranjan Padi, who worked under the chairmanship of Dr. Rakesh Bharadwaj, and he has selected 120 diverse cowpea germplasm lines, and the variant lines showing variability in the seeds, color, shapes, and he has done the biochemical parameters of all these 120 accessions of cowpea, and has found that there was negative correlation between starch and protein, phenol and starch, whereas positive correlation was found between sugar and phenol. And then development of NIRS based prediction model, which was his second objective for quality traits. He has done it for 4 quality traits: sugar, phenol, starch, and proteins. Successfully, he has developed for these 5 parameters, and this NIRS model can be used for rapid screening of large accessions in future. And some of the promising lines which he has identified for high starch, low protein, low starch, high sugars, low phytate, and high phenol. The second student, Mr. Chetan Kumar, worked under the chairmanship of Dr. Kavita Gupta for the assessment of genetic diversity and differential reaction of cowpea germplasm to pulse beetle. So he has evaluated the different accessions under artificial infestation conditions also, and has done the assessment of genetic diversity in cowpea using PCR-based molecular markers. And his collections were from exotic collections also, and the indigenous collections also. And based on the growth index, out of 83 accessions which he studied for their reaction to Calosobrochus chinensis, 4 accessions were found highly resistant, 15 as resistant, 39 moderately resistant, 19 moderately susceptible, and 5 accessions were susceptible. And based on the molecular markers, he has found that out of 26 SSR markers, 4 markers have 4 primers have given good diversity of the accessions, and compared to the. So main conclusion was from his study that the accession which he studied, the resistant accessions which I have identified, they can be used in the breeding programs for developing resistant cultivars. And of the 26 polymorphic primers, 4 were most efficient in classifying the genotypes into different clusters. And these can be used further in the breeding programs. Third student, G.J. Abhishek, he also worked on cowpea germplasm under the chairmanship of Dr. V. Celia Chalam, and he used different techniques like electron microscopy, ELISA, RT-PCR to identify and detect the BCMV using these different techniques to identify tolerant and resistant lines under the field conditions as well as in the artificial inoculation with the BCMV. So he has selected 85 cowpea germplasm accessions and 5 accessions each from 17 different states and 2 union territories. This is the distribution from where he has collected. He has field screening was done at ICR NBPGR in the new area as well as at the Ranchi regional section. And DEC ELISA was done for leaf samples of all 85 accessions and 1 susceptible check and 1 resistant check. And using all these techniques, he has confirmed the presence of BCMV, and the electron microscopy revealed that there are flexuous viral particles under the TEM. You can see in the photograph. And therefore, his main conclusions were the presence of BCMV was confirmed using all these techniques. Then there were BCMV specific primers were designed using bioinformatic tools, and the RT-PCR was standardized. Of the studies, 24 accessions, those were not showing BCMV infection in the field and in the artificial inoculation, and these accessions can be used further in the studies. And then there was no geographical D bar for the BCMV. The 4th MSc student, Mr. Venugopal Gowda, he worked with Dr. S. Rajkumar, and he studied the molecular and cytogenetic approaches to resolve the identity of vegetable Amaranthus species complex and to resolve the taxonomic ambiguity in the Amaranthus genus. And he has used cytogenetics and molecular approaches, morphological, as well as the sequence of nuclear ITS. They were used in 30 accessions which were non-designated as species, and based on his findings he can verify, he can identify that the tepal shape in the flowers can be used as a mark for the taxonomic identification in the Amaranthus tricolor and Amaranthus. And then variation in chromosome number, it could not be identified between and within the species of tricolor and the plant. And then consensus ITS sequence as DNA barcode was generated for identifying the Amaranthus species. Now I'll narrate the PhD students' work. First PhD student, Shailendra Kumar, he worked with the chairmanship of Dr. Sunil Archak, and he has done the analysis of genetic variation in Atrocarpus hirsutus, the wild jack collections from West. So he has gone for the collection in the central Western Ghat areas, especially in the Khan forest. 13 sites he visited and collected accessions from 8 populations. 63 trees were tags for the collection of fruits and leaves. Then the biochemical profile of wild jackfruit seeds was also comparable with the jackfruit and breadfruit, and you can see the different the collections. They are not very not as good as breadfruit, but they can be used. This is again the biochemical profile of wild jackfruit across the population in different populations. So his conclusions are that 30 cross-specific polymorphic SSR markers were identified, and they were used to analyze 56 genotypes, and they were classified into 8 subpopulations. Then among the 8 population 6 was distantly related to the others based on the Upma clustering pattern. Then Shefalika Amrapali also worked under the chairmanship of Dr. Sunil Archak, and she worked on olfactory, biochemical and molecular profiling of rose germplasm. She selected 205 rose germplasm and have screened for 27 fragrant cultivars shortlisted. And for the different chemicals analysis for fragrance, she identified 226 molecules were present, and basically they were of the terpenoid derivatives which were mostly abundant in the plasm. And then this is the analysis of her work. Then conclusion she made that high polymorphism was observed in all the 205 rose germplasm accessions, and no grouping was based And there was no grouping based on any horticultural classification or presence or absence of the difference. Third, Mr. Badal Singh, he worked with Dr. K.C. Bhatt, and he investigated the seed biology of Indian CWR of Rosaceae family from Himalayan region. He had gone 2 explorations in the Niti Valley, Nanda Devi Biosphere in Chamoli, Uttarakhand area, and he has collected many germplasms. And of these, 27 CWR, that is the crop wild relatives, belonging to 19 Rosaceae family, he has studied, and 33 of these have been successfully conserved in the gene bank. And this is the different prunus species which he collected from the area during his expedition. This is the species variability in prunus from Uttarakhand. This is again morphological variability in wild species. Then these are some of the basic studies like TTC, embryonic axis variability among different wild prunus species. Then for the first time, these seed germination protocols were standardized in the wild species. So the conclusion is that of the 45 accessions belonging to 15 wild fruits, which he collected in 2 explorations, 33 have been conserved. And this is the first report for the distribution of Prunus mira from Niti Valley. Earlier it was considered as Prunus persica. Then a new fruit species P. Prunus jackmontii, it has been identified for a direct domestication because it has good fruit bearing, good taste, TSS 22 and 46% of oil. And this I have already told that seed protocols were standardized. Then Pawan Kumar Malav, he worked with Dr. Anjula Pandey on morphological, molecular and biochemical variability of Ocimum tenniflorum, and he has undertaken studies on 109 accessions, 83 from gene bank and 26 from his own collections, representing 20 different states. And he has grown the crop in at 10 BPGR, and the analysis based on different parameters have shown that there were 8 different clusters which were present, and the variations within the population was maximum, different populations. Then GCMS profile of different chemical compounds, and the clusters, there were 7 different clusters were made based on the qualitative and quantitative parameters. And his conclusions were that diversity analysis revealed rich genetic diversity. Then some of the accessions have peculiar compounds which can be used for pharmaceutical industries. Then 2 different populations were identified based on extent of genetic diversity using molecular markers. And then gaps for collection, like from southern part, wild germplasm from southern parts and northeastern region, it needs further strengthening of collection. And then PGR student during the COVID period, because they were at their homes, they were given assignments and they have collected data and samples for their research program. And then finally, one student was placed, and then we have 33 total students on roll, 23 PhD and 10 MSc. Faculty strength is 6. And last year, our earlier professor, Dr. Rekha Chaudhary, she superannuated on 30th of April. And after the COVID was over, the farewell was arranged on 31st of January this year.
Thank you, Dr. Veena, for a very good presentation. The next presentation is by Dr. S.K. Chakrabarty, Seed Science and Technology.
Now, in the significant educational achievements for 2021 pertaining to the Division of Seed Science and Technology, with the mandates of strengthening the human resource development in seed science and technology, besides the development of seed production technologies and basic strategic and applied researches, we have the faculty of 34 faculties with 23 research guides, and presently we have 46 students. Now, in the year under report, there are 6 MSc students and 2 PhD students have earned the degrees. In the students' research programs, the first theme is the seed production technology. The quality seed production in cucumber has been studied to to determine the physiological maturity, harvest maturity, and fruit retention, to study the effects of foliar applications of nano zinc and nano iron oxides on the seed quality and yield associated with physiological and biochemical changes in the foliar applications of the nanoparticles. The results indicated in the seed development and maturation that the seeds attain physiological maturity on 40 days after uh, pollinations and 50 days after pollinations in the open field conditions and protected conditions. The fruit load per vine studies has indicated that the retention of 2 fruits per vine showed the higher seed yield and better seed quality in the open field conditions. The fruit positions also similarly has shown that the middle and styler segments showed the higher number of field seeds, 1000 seeds weight, and germination and vigor as compared to the peduncular segments of the fruit. The results on the foliar applications of N nanoparticles and its seed quality showed that the 200 ppm nanoparticles of iron and 300 ppm of zinc nanoparticles showed the better quality attributes irrespective of the varieties, seasons, and environments. Now, in the seed quality evaluation theme, we have two research theses. One is on the reactive oxygen species dependent seed germination mediated by NADPH oxidase, where the activity of NADPH oxidase and reserve mobilization has been studied in different types of mustard varieties. The results indicated that the lowest seed vigor index 1 and 2 in the 00 mustard genotypes with the maximum EC values. Now, the decrease in starch content started first, followed by the decrease in lipid and almost no significant decrease in the protein content. Maximum reduction of lipid content was observed in the 00 mustard varieties. The results further indicated that the NADPH oxidase activity was found to affect the post-germination growth and root development. It is one of the very strong source of superoxide production. Hydrolyzing enzymes, namely amylase, lipase, isocytate and protease, are associated with the seed reserve mobilization, and they are shown to have a very good correlation with NADPH oxidase activity. Similar studies on the mitochondrial and glyoxylate cycles metabolism has also been studied in Indian quality mustard seeds that again showed that the SDH activity and IDH activity was positively and negatively correlated throughout the time of germinations respectively. And a higher concentrations of IDH activity was shown to be the point in quality mustard types germination. And the results also showed that the lower malate synthetase and higher ICL activities had a poor seed germination and vigor in Indian quality mustard varieties. SDH is a direct source of ROS, which is involved in germination and plant development. So low SDH leading to low ROS, causing very poor germination in 00 mustard. These two studies has showed that the 00 mustard's poor performance in the germination and vigor is because of the ROS activities and other associated enzymes. In the seed quality testing, the phenol color reactions in the rice seeds has been studied with the objectives to standardize the phenol color testing method, to understand the occurrence of phenol color reaction, intra and inter varieties phenol color reaction assessment, and biochemical basis. The results indicated that either 5 ml of 2% phenol in 11 centimeter petri plates or 4 ml in 9 centimeter petri plates kept in 4 hours in a temperature of 25 degrees centigrade plus minus 1 showed a uniform color development across the 5 different color groups with black, dark brown, brown, light brown, and no color. In order to understand the induction of phenol color reaction, pre-anthesis and post-anthesis stages plants have been identified, and the results showed that the phenol color does not occur at the pre-anthesis stage, where post-anthesis stage phenol color occurs. The biochemical basis on phenol color reaction has shown that the mean phenol content, pyracatechol activity, L-dopa activity, and tyrosinase activity has shown a very increase in the activity along with the different phenol content from no color to dark color varieties. However, the 1-year-old seeds showed a higher percentage of phenol color reactions along with the higher content of polyphenol oxidases. In seed quality enhancement experiments, the 4th thesis have been performed, And in the first one is in the pulp millet on nanoparticles, use of nanoparticles on seed germination and longevity. This particular research has shown that silver nanoparticles at the rate of 50 milligram per liter enhanced seed germination and vigor in terms of radical emergence and vigor indices in the parental lines and the PUSA composite 5701 in the pulp millet. Similarly, in the objective to increase the seed longevity in pulp millet varieties, the results showed that silver nanoparticle primed seeds had a higher seed longevity in terms of higher germination and vigor as compared to the hydroprimed and unprimed seeds. However, with extended period of aging or storage, these priming benefits are lost. In onion, the nanoparticles have been used particularly against the spottle blotch disease, and the zinc oxide and graphene oxide has been tried in different concentrations. The nanoparticles have been characterized using UV spectroscopy and Fourier transform infrared spectroscopy. The results indicated that zinc oxide nanoparticles at 100 and 250 ppm significantly improved the seed quality and significantly increased the higher activity of catalase, peroxidase, and ROS activities. The antifungal activity of both the nanoparticles have been studied here, and the zinc oxide nanoparticles were 100 ppm was at par with the commercial fungicide SAF. However, the graphene oxide nanoparticles enhanced the seed quality, but it could not be found effective against the seed-borne pathogen control. In use of nanoparticles on soybean for seed quality parameters and its seed yield and its contributing characters evaluation using zinc oxide, titanium oxide, and silicon oxide of different concentrations, both in bulk and nanoforms in two varieties, showed that the highest germination percentage was obtained by zinc oxide nanoparticles at 500 ppm, followed by silicon oxide 700 ppm and titanium oxide 500 ppm. Now, these concentrations have also been tried in the field conditions, and there it is shown that nanoparticle zinc oxide 500 ppm followed by bulk zinc oxide 700 ppm enhance the seed yield parameters for both the varieties. In maize, in sweet corn maize, in the parental lines and hybrids, in an idea to see the effect of nanoparticles for low temperatures mitigation, the research has shown that the under lower germination and vigor at low temperatures have been obtained, and higher germination and vigor in the hybrids as compared to the parental lines, both in the germination percentage and the vigor indices. And lower enzymatic activities are also found in the low temperature germination conditions. In order to mitigate these effects of stresses, the hydropriming, halopriming, solid matrix priming, and magnetopriming has been tried to whether it can be mitigated. Now, the results indicated that hydropriming and magnetic treatment improved all the seedling parameters. Particularly, it improved the root growth and its associated characteristics. Now, the faculty has been honored and awarded and recognized for different activities. Dr. Monica Joshi has got the Outstanding Woman Scientist Award, and Dr. D. Vijay, Dr. Atul Kumar, Dr. Tejpal Singh has also obtained different awards and prizes for best oral presentations and poster presentations. Among the students, Bal Chandan Gowda and Dilsad Ahmed has also been awarded with best poster presentations. Now, the students of the division has been obtaining the fellowships awarded by UGC and ICAR. Now, one international training, one national training has been organized in the division in the year under report. The publications consisted of total of 28, including 12 from the students' research. Few equipments have been purchased for postgraduate research program. I acknowledge the help and support provided by the Director, Dean, and Associate Dean of ICAR-IRI, the students, faculty, and staff of the discipline of Seed Science and Technology, and the staff of PG School IARI.
Thank you. So we have come to the end of all the three presentations. The work done by 37 students were very nicely presented by three professors. Now I would like to invite the chairperson, Dr. Shailaja, for her remarks and further discussion. Ma'am, kindly unmute, ma'am.
Kindly unmute.
Can you hear me?
Yes,
Yes,
ma'am.
ma'am.
Okay, very good. We had all the presentations and the teachers, their supervisors present. Their work was very good, and the presenters covered it. Dr. Vinod and the seed technology and the genetic resources, they made it very brief and still covered entire research findings. That was very good of them. And firstly, I congratulate all the guides of all the three faculties who have given a very important research problems which are actually practical and useful for the farmers, research oriented and applicable. And IRI has this tradition of having farmer oriented research programs. And we can see from the story of Pusa Basumati, not only farmers, it has also been commercialized and benefits the institute. And I foresee even in these research problems, which I saw in plant breeding and some of them in science and seed science and also in the plant genetic resources, they have identified key findings which can be useful for various people and even the companies to be commercialized and directly to the farmers. So I congratulate all the guides, firstly of the students who have given such a important research problems for the students. That I think should be congratulated to the guide, because guide also spends a lot of his brain power in guiding the students, and also because of his or her experience, they find this research problems very useful. Then faculty wise, I want to make small comments on plant breeding. Basically, I think here there have been very good progress, because what I see is the choice of the crops are very diverse, and they are for the local issues of North India, like Haryana, Punjab, Delhi, and all that. The crops are very appropriate for that region. So the experiments have been taken in that region. And as a plant breeder, they have used various methods, not only the choice of the crops, they have used all the tools actually, both conventional, molecular, and up to the latest. I see the breeding methods, crossing, molecular, mass, GWAS, mutation studies, sequencing, and mapping, and all. So there is a host of tools they have used in identifying and addressing the research problem. So that's very good. And right from cereals, millets, and cruciferous crops and so on. So all areas are covered and address the problems of the farmers. Then they have identified key products also at the end using, and they have used, and I think they have good facilities to carry on this research. And they have also used good tools actually, and methods and systematic way of planning their experiments and getting the products for that. It's as though it's a students are trained well to become very good scientists or good agriculture researchers. Then we have, this is totally product oriented. So we can foresee the products for which the issues have been addressed. We can see there are markers, there are crops, there are genotypes, and variety, leading to varieties. So they have issues, and especially they have anthocyanin in the rice that was done in the Northeast. You can see they have identified this black rice has so many importance, and nitrogen use efficiency, then the disease resistance, introgression breeding, everything is there. So I feel very good that the products have been identified from various crops. Every thesis has something to have a product. So that is very good. And lastly, the big point where we see everywhere is the publications. But I see here there are good publications. So it is a good effort from the guides to see that the students' research is published and in a very high rating journals also. So that is very good point, because most of the guides, as guides, we have failed to make the student write papers of all their works that they have spent. So here they have produced papers in very, very high-end journals, products, varieties, and so on. So I feel in the plant breeding group, the guide and guides as well as students, they have spent time to have a very good thesis, and I'm sure many of them would have been awarded also. And I congratulate all these students and teachers. All in every faculty, teacher and student has worked very hard to do it. Then coming to the plant genetic resources, there has been a very good diversity analysis in all the crops they have addressed. And as we know that plant genetic resources, because of diversity, unless diversity is known, we can't select good parents. So they are all interlinked. In the breeding program, we need very good type of parents, how diverse parents or whatever. And here also they have used very good tools, and then they have identified virus resistant varieties, and then they have used in the breeding program, and we can select good. And one very good point I see in plant genetic resource, there was a thesis who has worked on cytogenetics. Cytogenetics has become a very, we can say it's on the way of extinct. Very few people are working on cytogenetics now. And they have used cytogenetics, though they could not get a very good result at the end, but they have attempted. So that's one I noted. And there have been pioneering efforts in plant genetic resources to find the wild relatives of breadfruits and jackfruits and so on, even though it's not the crop of that area, and diversities in rose families and wild relative of Rosaceae of Himalayan region. So the student has put an effort to go to the Himalayan regions, expedite, collect them and everything. So all these genetic resources has got a very good yield, and they are also practical oriented, and the guides have also put in a lot of efforts to do so. Coming to the seed science, the next faculty, here an effort of all the thesis is to find out the quality of the seeds, high quality of the seeds, how to identify them, because the initial thing for any good crop is high quality seeds, and they have used nanoparticles. The recent means in last few years they have been using nanoparticles, especially silver and zinc particles, to identify good quality seeds, and these are also very practical in use. So they affect germination, yield, disease resistance, and ideal temperatures for germinations. And also they have, the guides and the supervisors have used practical approaches for their students, MSc and PhD, to identify how very good seeds could be in different crops. So, so this is about mine, and publications also are good. So in all the three faculties, I think there is compulsion also to publish paper in every university. And here also they have done good publications are there. So overall, I want to summarize that in all the three faculties, there has been very good approach, planning and implementation for the postgraduate research. And they have also got good products. Some are end products, some are intermediary. So I only would like to have one suggestion for everyone, that whatever material has been developed by the students, they go away, they take a job and go away, but that should be followed up and lead towards either varieties, commercialization or something. So that, because so much money has been spent, time, effort, and the brain power of the supervisor. So all this has to be taken forward so that a reliable and appreciable product can be produced from postgraduate research. Because many times we have this misconception that products has to come from the research programs, the regular research program like ACRIP or university research or institute research. But the products of students research also is very valuable, and that can be taken forward by the scientist. And I'm sure many are doing it, but somewhere there is a weak point that some when the student leaves, it is forgotten. So this, and every research outcome should be published, not just one or two, no? I don't know, maybe in IRI they have a good tradition, because IRI is a very prestigious institute and high quality research has been done. So this has to be carried out. So overall, I congratulate all the graduates, the awardees, the gold medals, and best for that. And I thank Dr. A.K. Singh, head of the IRI, and Rashmi Agarwal, and other team members who have made it possible to happen. And I thank. So these are my few words of appreciation and suggestion.
Thank you, madam.
Thank you
very much.
ma'am. So,
Madam,
so nice.
madam,
Yeah.
Yes.
now we have a rare distinction of having Professor R.M. Singh, sir. So before our director comes in, it is the elder has to take the stage now before the younger one comes. So now I request to Professor R.M. Singh, sir.
Before he shares his impression about what has happened, I would briefly like to say, for the benefit of other 564 participants who have joined us offline, Professor R.M. Singh is a distinguished teacher. He did his master's from Pather College then, and then PhD from University of Florida, Gainesville. And he has been one of the classical cytogeneticists. Still, his cytogenetics, pictures that you see he has done for his PhD program, they are landmark and always something good to see. And we aspire to have that kind of slides. Dr. Shailaja mentioned about cytogenetics getting extinct, and they are the people of the time who did great pioneering work in cytogenetics in Avena, and then in mutation breeding, in disease resistance breeding, and his contribution in human resource development in field of genetics and plant breeding is monumental. You name who is who in field of genetics and plant breeding, and they have been all his students, whether it is Dr. Prabhu, N.K. Singh, myself, Joshi, Ravi P. Singh, and many more. They have all been students and taught by him. So, and it was on his sheer interest that he wanted to see what is happening in IRI in terms of science. We keep discussing. He always keeps poking me for doing, innovative things that others are not doing, and it was sheer his interest that he's here. So. Would be happy to listen from him about his impression, what is happening in IRI and how we should follow. Over to you.
Thanks, Dr. A.K. Singh, for such a nice introduction. I don't know whether I deserve all that, but I appreciate it very much. Let me, at the outset, express my gratitude for providing me this opportunity to share some of my views with you all. All the three disciplines, crop improvement, the genetic resources, and the seed science, I can see in crop improvement very good presentations. The outcome has been very significant, and most of these researches have been published in very high rated journals of international and national levels. So I congratulate all the awardees as well as the guides for their efforts to have such a nice group which has been working together for the cause of science and ultimately the society. Similarly, genetic resources, as has been said by the chairperson here, that is very important for any crop improvement program. And we first look for the germplasm where the trait of interest is available there, or not. If it is available, we go straight away and use it in our crop improvement program. If it is not available, then we follow a number of procedures like mutagenesis and others to induce some of these traits of interest so that these can be used. And genetic resources, I could see that some very good work has been done. And this work is of quite great importance, and the publications have been also brought in there in good journals. In seed science, the major emphasis I could see was that how the seed quality is improved by using various nanoparticles, particularly of zinc, I could see. And there was a significant improvement in the germination of seed, and thereby you can use it. So all these researches have definitely led to some new ideas, new innovations, and for those, the faculty members, definitely they have the credit which goes to them. After listening the presentations, I have a few queries maybe, and that is particularly what is the evolutionary significance of phytic acid contents in various crops, which I could see that a number of crops have been taken and phytic acid content has been studied, particularly low phytic acid contents. There was a mention of a mutant. I don't know whether it is a naturally occurring mutant or whether it is induced mutant. When you have high phytic acid content, what is the significance of it to the plant itself? That is a very important point in my view. That is one thing. The second one, in the black rice from Manipur, I could see some very good lines developed using a Pusa dwarf material and the tall black rice, and some one was working at IRI. I had interacted earlier also on that, and here I could see that some dwarf lines have been produced in this. What is the character-wise, the quality-wise, as compared to the original material, how they stand, where they stand really? And third point, that in 000 mustard, where erucic acid and the glycosinolates have been removed, the oil is good, the percentage is, and the quality definitely is good, but the seed size is very, very small. And the farmers, I had some material at Varanasi, planted it in various farmers' fields, and their reaction was that when we go to the market to sell it, they don't get the remunerative price for it. And therefore, whether some improvement in the seed size and its color has been made, having that quality of it, the 00. So these are a few remarks as far as the crop improvement are concerned. With that, I would like to again thank Dr. A.K. Singh for giving me this opportunity, and all the other members, particularly the chairperson, for a very nice summary of the entire presentation of the discipline. Thank you very much.
Thank you. Thank you, Professor Singh. Dr. Vinod, if you permit, because these questions belong to my two PhD students directly, so I can briefly
This is
tell
your
about
residence
those
work. Yeah.
two, and also yeah, the second one. So the first one is about phytic acid, and phytic acid acts as a chelating agent for the micronutrients iron, zinc, and this is an anti-nutritional factor. So when phytic acid is present in higher quantity, it chelates with iron and zinc, and therefore its bioavailability in the human system is drastically reduced. Therefore, our efforts is to breed varieties which have low phytic acid. So in that context, we started this program, and this mutant was generated as a part of our tilling project, where we had EMS-induced mutagenesis in the background of Nagina 22 variety of rice. And from there, this mutant, large number of mutants were generated. And when we started screening this, we identified this as a low phytic acid mutant, and then we went for detailed characterization, mapping, tagging, and then our efforts is to transfer this to other basmati, non-basmati varieties through marker-assisted breeding, because now we have a marker available, so that the bioavailability of iron and zinc in these varieties can be enhanced. So that's the basic purpose. Phytic acid, when we reduce too much, it does have some effect on plant growth and development, particularly germination. And therefore, we have to have a balance to what extent it should be. That's on the first issue. The second PhD student, the first one was done by Prem. Now he has joined as a scientist in Forestry Institute. And the second PhD program was one of my in-service candidate student. She has Bhuvaneshwari. She has worked on black rice, and this is called Sakhao Paritan from Manipur. This is protected under geographical indication. It's a high-value commodity, and I understand in near Varanasi in Chandauli region, Prime Minister made his statement in Man Ki Baat. Some farmers have started growing in large area black rice from Manipur, and they are getting 800 rupees per quintal price for that. Of course, it is protected in the geographical indication, and it has to be cultivated in Manipur area to be called as black rice. But these are traditional tall varieties, very low yielding, and the major focus was how we can first characterize it at molecular level, biochemical level, what makes it having so much of antioxidant potential. So that aspect has been studied. And then the idea of bringing dwarfness from one of another source of dwarf mutant that we had in the background of variety Vindali mutant 68, that was used as the original source. And now this Sakhao Paritan, which is very tall type, about 1.5 meters, now we have made it dwarf to less than say 90 centimeters. The idea is to avoid lodging, enhance its productivity. And you rightly said that the quality has to be maintained, and therefore we have taken all biochemical parameters in terms of characterizing the anthocyanin, the different components of anthocyanin, and their antioxidant potential in the original Sakhao Paritan and the derived lines. So on those parameters, it is perfectly matching. And we are trying to see that now these dwarf lines, they become available in the Manipur region. I am trying to talk to the institute in those regions, ICR institutes, that we can go for large scale. It's the release and then cultivation in large scale. So that's about the black rice. And of course, third question that was about the small seed size of the 00 mustard varieties. Earlier, we had said Pusa 00 mustard 31, which is yellow seeded and a small seed. That's rightly so, and farmers do not get price when they go to mandi. Of course, they can have their own oil extraction and then good quality oil, but they have to go to mandi also. So considering that factor in mind, the new series varieties, now this year, a new variety has been released from CBRC, is Pusa Mustard 32, PM32, which has got bold seed size and brown seed coat color like normal mustard. And its seed size is about 5 to 6 gram, which is as good as Varuna or other varieties. And that is single zero, not 00, it's a single zero, PM32, PM30, and PM29. These are three varieties which are single zero. As far as oil is concerned, it is zero erucic is important. So these varieties would be replacing this one. And then it is little difficult task to combine the 00 with this seed size, and that is our major focus. Our mustard breeder, Dr. D.K. Yadav, and then his colleague Naveen and all other people are working intensively. Thank you very much.
Thank you, Dr. A.K. Singh, for explaining all the aspects which I has raised. Still, the first thing which I said, that phytic acid content evolutionary significance, why it was high in the initial material. I mean, all the material which you see there, it was very high. What was the significance of that high phytic acid contents to the plant itself? Was it resistant to some diseases or some other aspects? Some of this thing. I thought that there must have been some reason for its natural selection.
Right, good point. We will explore more on that. Not very planned information or investigated information is available, but it has a relationship with the general stress tolerance in these lines. And also at the time of germination, it helps better germination. Because we have seen that the lines which have reduced fatty acid in case of soybean, in case of maize and other crops, rice also, they have tendency to have reduced.
Okay.
Dr. Veena Gupta wants to add something from DG.
Good morning, sir. Nothing special to add, but I would like to thank for their encouraging words, Dr. Shailaja Madam and Dr. RM Singh, for the cytogenetic work which my student has done, and for the wild crops, another student Badal has collected from the Uttarakhand area, which is now in a very dangerous condition because of the glacier there. So I would like to thank you, sir, for your encouraging word, and we would like to carry forward this work again with other students, coming students in future. Thank you, sir.
It's very important really, because we see that cytogenetics is disappearing from the scene. When we look back, Sears period, you can see that how that gene was brought on a segment of translocated in case of wheat, and that revolutionized the whole wheat cultivation. So, I mean, such things are definitely very important. And if one uses this as a tool and try to really take other tools together, definitely some very useful things can be generated out of this discipline. And therefore, I think there is a need for this thing, strengthening this thing, and people should come forward and take. And as a matter of fact, the scientists who are guiding PhD scholars, they should also do this thing, give some problems of importance where it can be of direct use. Thank you very much.
Thank you, sir. Thank you.
May I add something here, sir, if permitted?
Sure.
Yes.
Yeah.
Yes.
Actually, we are using chromosome manipulation techniques in wheat. And if you see the very first presentation where we transferred a gene from agilosomography. It is a tertiary gene pool species with C genome. And we used pH1 nullify B mechanism to transfer this gene. So cytogenetics and chromosome manipulation techniques were extensively used in genetics division for transfer of rust-resistant genes from wild species into wheat.
Very good.
Okay.
Very good. That's,
Very
I'm
good.
happy to know that.
Thank you. Let us move forward.
Yes.
I think we have Professor H.V. Singh, so we can start with the next group of presentation.
Yes, sir.
Dr. Rasmi?
Yeah, Seema Jaggi will take
Seema,
over.
please.
Before we start the next session, I would like to extremely grateful to respected Dr. Shailaja for accepting our request and chairing this session. Your remarks, suggestions, and comments for all the three disciplines are very valuable for us. Thank you for appreciating and encouraging the efforts and work done by the students of this institute. Thank you so much, ma'am.
Thank you.
Thanks to Professor RM Singh, sir, for being with us and for appreciating the efforts of the researchers, highlighting your observations and blessing us on this occasion. Thank you so much, sir. Thanks to Dr. A.K. Singh, sir, and Dean ma'am. And thank you so much to all the professors who made their presentations. It was excellent presentation. Thank you so much. And thanks to all who have joined this session. Thank you all. I think we'll continue, sir.
We'll continue the session. We will have next Yes,
Okay.
Seema.
Okay. Thank you.
So we would like to continue the session, and this second session is on the presentations of the significant educational achievements made under the School of Crop Protection. So with a brief introduction, the School of Crop Protection includes agricultural chemicals, entomology, plant pathology, and nematology. So there are total 41 students who are going to get degree this year, and which includes 19 MSc and 22 PhD students. So the significant achievements of these 41 students will be presented by the respective professors of the various disciplines. And at the outset, I would like to welcome Honorable Dr. Harikesh Bahadur Singh, an eminent educationist and researcher who will be the chairing this session on School of Crop Protection. So at present he is a distinguished professor, University of Biotechnology, GLA University, Mathura. And before this prestigious position, he served at many various prestigious positions, and also he superannuated as head Department of Mycology and Plant Pathology, Institute of Agricultural Sciences, Banaras Hindu University, Varanasi. So, sir, we heartily welcome you, Dr. H.B. Singh, and he has immensely contributed in the field of sustainable agriculture through identification, characterization, and field validation of biocontrol agents for the management of various diseases. Sir, I would request our director, Honorable Director, to introduce the chairperson of this session, Dr. H.B. Singh.
Thank you, Dr. Rasmi. You have, of course, given introduction of Professor Singh, so I have nothing much to add, except that I have had a great admiration for Professor H.B. Singh when I was doing my BSc Agriculture in Banaras University. He was at the verge of submitting his PhD thesis with Dr. U.P. Singh in Plant Pathology Division. And I used to see him working so intensively many times in the campus of Vishwanath Temple, right? We have in the university campus front of Institute of Agricultural Sciences. He will be doing his research papers and thesis writing, sitting in the, the temple campus many times. We used to see him from a distance, and it used to be a great admiration. Dr. Singh has done monumental research work in field of fungal pathology, and he worked at CMAP and NBRI and at Banaras Hindu University. He's fellow of National Academy of Agricultural Sciences, and his major focus of research has been in the area of biopesticide, biofertilizers, biostimulants, and nanotechnology, organic farming. And he is ever young person that I see, always full of energy. He has been regularly attending all the NAS Academy meetings very regularly and contributing to the discussion program. So I would like to welcome from my side Professor H.B. Singh to this program to chair this important session on the School of Plant Protection disciplines. Thank you.
Dr. Seema, you can take.
Yes. For wonderful introduction. You introduced me so nicely. I'm grateful to you that you gave me an opportunity to chair this session, very important session. Of course, I was associated with IRI since very long period. And only last week we conducted a PhD Viva OC of Dr. Rashmi. So I think without wasting time, we can straightway move to presentation, Rashmi.
Yes, sir. Yes, sir.
And we will have
yes, sir. Subsequently, we'll discuss, sir. Dr. Seema.
Thank you, ma'am. In this session of School of Crop Protection, we have 4 presentations from the disciplines of agricultural chemicals, entomology, nematology, and plant pathology, wherein work of 41 students will be presented. So the first presentation is by Professor Neera Singh from Agricultural Chemicals.
Good morning. I am here to present significant educational achievements by student faculty of discipline of agricultural chemicals. Mandate of division include development of agrochemicals, their formulation, safety evaluation, and human resource development.
Is very low.
At present, we have 16 faculty members, 34 students on roll, and 2 have taken relief. Devabrat Ghoshal, Harshak Kumar, Kirti Raj, Shirosi Biswas, and Partha Chandramandal will be getting their MSc degrees. Samrendra Singh, Dinesh Kumar, Niranjan Kumar, Shubham Yadav, and Usha Kumari will be getting their doctoral degrees. Students have addressed theme areas of development of agrochemicals, formulation, safety evaluations, and decontamination by their thesis. Kirtiraj did bioprospecting of Pogostemon, commonly known as Patchouli, for its insecticidal activity. Essential oil and leaf extract were evaluated for acariciidal activity. Among the extract, methanol extract was highly effective, while essential oil also showed significant activity. The characterization of hexane and the essential oil by GCMS suggested that pitcholol was the main constituent present in both fractions. Essential oil and hexane extract were used for preparation of coarse and nano emulsions. Acariciidal and antioxidant activity evaluation suggested that nano emulsions were better than the coarse emulsion. So study conclude that Pogostemon has potential for its use in plant protection. Partha Chandramandal evaluated eucalyptus and Simbopogon, that is lemongrass, for the control of Bemisia tabaci, that is whitefly. Essential oil was extracted from eucalyptus and lemongrass. Then pure constituents were isolated from the essential oil and the acetone extract of eucalyptus. 1,8-cineol was main constituent in essential oil of eucalyptus, while citral was main constituent in lemongrass. The toxicity evaluation suggested that essential oil of lemongrass and eucalyptus extract were most active against Bemisia tabaci. Ovipositional deterrence assay suggested that at 0.05% concentration, both essential oil were effective against whitefly. This study concluded that eucalyptus extract and lemongrass oil were effective in killing whitefly, while essential oil possessed ovipositional activity. Dinesh Kumar Yadav evaluated ferrucinile chalcones for their phytopathogenic fungi and root knot nematode control in tomato. 34 compounds were prepared following the methods shown here. Microwave method was better than the conventional method as far as the preparation time and yields are concerned. Synthesized compounds were evaluated for their fungicidal activity. They possessed significant activity against Sclerotium rhamnosus and Alternaria solanae, but activity was less than the standard compounds used for their control. In vivo studies suggested that drenching method was better than the root dip method, and at 1000 ppm concentration, one compound 3B possessed best activity. Compared to fungicidal activity, nematicidal activity of ferrucinile chalcone against Melidogyne incognita was better. The in vivo evaluation suggested that compound 3F at 80 ppm concentration showed activity at par with carbofuran, and drenching method was better than the root dip method. So these chalcones are promising in disease and nematode control in tomato. Shriyosi Biswas prepared a novel premix formulation of chlorimiquate chloride and tebuconazole for lodging prone crops. Premix formulation was prepared and evaluated under phytotron in field condition, and bioefficacy and residue data was recorded. The in vivo field testing results suggested that the prepared formulation was better than the tank mix formulation as far as plant height is concerned. Grain yield was better in the premix formulation than the control and the tank mix. So the premix formulation of chlorimiquate chloride and tebuconazole showed supremacy compared to individual application or the tank mix formulation. Devavrat Ghoshal prepared imprinted polymers for the removal of thiomethoxam from complex matrices. 15 MIPs and 15 NIPs were prepared and optimization of the binding parameters for best activity was done. The binding specificity for thiamethoxam was more than 90%. They can be regenerated and reused and showed good recovery from honey, brinjal and rice samples. So prepared MIPs can be used as an adsorbent in the SPE cartridges. Harshan Kumar evaluated fate of Pusa hydrogel and its variant in soils. LC-MS for quantification and Quetches method for extraction was standardized. Then persistence of both gel in laboratory and in field was studied, and results suggested that none of the monomer or cross-linker was detected above the limit of quantification in soil or commodity. Adsorption, desorption and leaching studies suggested that acrylamide and methylene bisacrylamide was poorly sorbed in the soil and showed leaching up to 70%. This study finally concluded that monomer and cross linkers were never detected in the soil and commodities, but these monomer and cross linker they are poorly retained in the soil. Niranjan Kumar studied fate of cyanotraniliprole in 6 different soils of different agroclimatic regions of India, and both laboratory and field studies were performed. 6 soils from different agroclimatic regions of India were used in the study, and the half-life values varied from 13.1 to 38.5 days. Effect of abiotic factors, amendment and biotic factors on degradation of insecticide was evaluated, and all these factors played a significant role in degradation of insecticide. Leaching studies were performed in packed columns, and soil varied in their potential for insecticide leaching. 96% of added cyanotraniliprole was recovered from the soil, while only 4% was recovered in the leachate. Metabolites of insecticide were more mobile and leached up to 25 cm soil depth. Field evaluation studies were done in IRI soil. Compound was degraded after 5 days both in cabbage and in soil, while the leaching study suggested that cyanotraniliprole was present up to only 10 cm depth, while metabolites leached down to 25 cm depth. So parent compound is safe for its use, but metabolite has concern of leaching. On-farm burning of sugarcane trash is practiced. Therefore, Shubham Yadav evaluated role of ash on fate of atrazine and fipronil, two sugarcane pesticides. Ash was characterized for its physicochemical characteristics. Then sorption was studied in 3 different soils from sugarcane growing area. Both compounds were sorbed more in ash mixed soils, but effect varied with soil type. Sorption was more and desorption was less in the ash mixed soils. Leaching results suggested that ash resulted in higher retention of atrazine in the surface 0 to 5 cm layer, while no effect on mobility of fipronil was observed. Degradation studies suggested that the ash resulted in higher persistence of atrazine in all 3 soils, while no effect on fipronil degradation was observed. Bioavailability studies suggested that the ash reduced the availability of atrazine in soil solution. Therefore, its availability for weed control was reduced. Finally, this study suggested that the addition of ash, which is a continuous process because farmers burn trash year after year, may result in increased persistence and reduced activity of soil applied herbicide. Samrendra Singh studied microbial degradation of PCBs and isolated microbes from different sources. Number of bacteria and fungi were isolated from paint scrap, cement debris, and transformer oil contaminated soils. Isolated microorganisms alone and in combination were evaluated for the degradation of PCB mix. The results suggested that the fungal strain isolated from the paint scrap site were the best in degrading PCB mix, and half-life values were reduced by 15 times as compared to the uninoculated control. Methoxy metabolites of PCB were detected during this. Usha Kumari evaluated biobed for atrazine and fipronil rinsate disposal using amended biomixtures. The mixtures were amended with ash or biochar and were inoculated with atrazine or fipronil degrading microorganisms. She prepared 9 different biomixtures and results suggested that the rice husk ash and biochar mixed biomixture were highly effective in absorbing these two pesticides. 7 biomixtures were evaluated for atrazine and fipronil degradation individually, and effect of concentration and bioaugmentation was studied. Except 5% WBC biomixture, atrazine and fipronil were easily degraded in all biomixtures. Bioaugmentation greatly increased degradation of both parent and metabolite. 3 best biomixtures were used to prepare biobeds, and degradation of atrazine and fipronil in mixture was evaluated. These biobeds significantly enhanced degradation of both pesticides, but effect on atrazine degradation was maximum. Both parent and metabolites were degraded. Therefore, these biobed can be used for on-farm disposal of pesticide waste. 31 research papers were published during the period, which included 70 student publications. Faculty members were recognized as members of different committees or Best Teacher Award and Best Presentation Awards. Arkadev Mukhopadhyay and Rakesh Kumar were awarded Prime Minister Fellowship, while 5 of the students have been selected in ARS. Finally, I would like to thank all the faculty members, research guides, and all students who contributed for this presentation. Thank you, everyone.
Thank you, Dr. Neera. The next presentation is by Professor Devjani Day from the discipline of entomology.
A very good morning to all respected dignitaries and dear colleagues, students, ladies and gentlemen. It is my pleasure to present before you all the significant achievement of the students who will be receiving their MSc and PhD degrees in this convocation. The mandate of the division is to conduct basic and strategic research, develop as a center of excellence in research, teaching, and human resource development to provide services in entomology. 13 students will be receiving their degrees, and from the various research themes, it is very clear that all the areas of entomology were addressed by the students. Authentic identifications are very important for success of biocontrol and IPM programs. Therefore, efforts were directed to study certain economically important groups. The first study was on family Delphisidae, an important plant hopper family. this work was done by N. Ramaya under the guidance of Dr. Mishram. 3 objectives were there for this study. A checklist for Delphicidae was compiled. It included 73 species under 26 genera. Explorations were made for collection of specimens from 26 localities under 9 states. More than 2,000 specimens were collected and they could be identified into 39 species. As is imperative, exploration led to establishment of new records for India. 13 species were recorded for the first time from India. She also described 9 species under 2 of the subfamilies of Delphicidae, just to show the amount of work which is involved in description and redescription of the species. These are illustrations which accompany each description. An important work which was done here was preparation of a diagnostic key for identification of the Delphicid complex in rice. Easy characters and good illustrations accompanied the key, and this can serve as a ready reckoner for rice workers. 32 GenBank accessions were generated. Targeted genes were histone, MTCO1, and 26S. The phylogenetic tree based on the sequences showed that both these subfamilies, tribes of Delphicidae, are paraphyletic. The second study was on lesser known tribes of Chiasmini and Paralimnini. These are leafhopper tribes. Again, Akash Nikoshe worked under the guidance of Dr. Meshram with the following 3 objectives. He could collect insects from 46 locations, and around 3,000 specimens were collected, and 37 species could be identified from the collection. 11 new locality records were established. A checklist for both the tribes were compiled. 6 new species were described, and another study was conducted on the leafhopper tribes Opsini and Coebellini by Sunil, under the guidance of Dr. Meshram. These were the objectives for the study. He could collect from 22 locations of 13 states. More than 2,000 specimens were collected and 34 species could be identified from the material. He could record 1 genus for the first time from India, Libin-Legea. This was recorded from Kerala and New Delhi. New reports of another 5 species was also established. 7 locality records within India were established. He could describe 2 new genera and 7 new species. These are the details of each of them. A molecular phylogeny of the tribes was worked on based on the genes histone 3 and 26S RNA. And it was seen that Opsini was paraphyletic while Coebellini was monophyletic. A 4th study was done by Prashanti under my guidance by studies on hymenopterans associated with major pests of rice. These were the 4 objectives. She compiled a checklist of 67 genera with 103 species under this. The checklist gave details of the stage of host and also the name of the host and distribution within India. Around 1,000 parasitoid specimens were reared, and they were identified as belonging to 60 species. Several first records were established. 13 larval parasitoids, 5 pupal, and 3 egg parasitoids could be recorded on Sarcophaga incertulus. Similarly, 6 larval parasitoids and 2 pupal parasitoids were found as new records on Ephalocrosis medinalis, and 3 nymphal and 2 egg parasitoids could be recorded on Nilla parvata lugens. 29 DNA barcodes of various parasitoids were generated. More than 10 of them were first submission in the database. The community structure of parasitoids vis-a-vis pest and plant stages was studied for 2 crop seasons of 2019 and 20. It was seen that species abundance, diversity, and richness was greater in the organic ecosystem compared to the conventional rice ecosystem. A study on Lepidopteran stem borers of graminaceous crops was undertaken by Adrish Dey under the guidance of Dr. Shashank. He has already presented 2 days back. 13 species of stem borers belonging to 5 genera were re-described with additional details and emphasis on male genitalia. Molecular studies showed that Indian population of Sesamia inferens is genetically homogeneous, but there is evidence of population expansion. Another study was by Shravani, again under the guidance of Dr. Shashank, on this family Sphingidae. These are commonly called as hawk moths. The study had 2 objectives. She compiled a checklist of 223 species belonging to 60 genera from India. She's re-described 29 species and augmented 11 species which were not there in our National Pusa collection. Mr. Venkanna Yana, under the guidance of Dr. Sachin Sudesho, worked on the predatory behavior of coccinellid beetle, Chilominus sex maculata, and its predation risk on prey. His work had 3 objectives. He standardized a technique for rearing cotton aphid in the laboratory on Hibiscus cannabis. And using his technique, it was seen that 27 degrees centigrade was more suitable for production of large number of nymphs and wingless adults, while 27 degree was found to be more suitable for production of winged adults. He conducted life table studies on Chilominus sex maculata also, and it was seen that mean fecundity of these beetles was 790.19 and mean generation time was 27.29 days, while the intrinsic rate of increase was 0.216 days. Feeding potential of sex maculata on Apis gossypi was also studied, and it was seen that 4th instar among the grub stages and female among the adult stages are voracious feeders. Lotka-Volterra model was used to calculate the total response of predator and change in the prey population, and it was seen that female beetles were the most effective predators, followed by the 4th instar grubs and the male beetles. Diversity and characterization of gut flora was studied by Mr. Kondwani in Lepidopteran and Coleopteran insects under the guidance of Dr. S. Subramaniam. The study had 3 objectives. He identified and characterized the gut flora from Eri silkworm, Samia ricini, and the grubs of Anomola dimidiata. The identified flora belong to 2 of the phyla, Firmicutes and Protobacteria. 60 aerobic culturable gut bacteria were recorded from silkworm, while only 40 could be recorded from root grub. And a comparison showed that some of these isolates, which are specific for each of these insects, showed less similarity, suggesting the possibility of novel ones. Culturable anaerobic gut bacteria were also isolated and identified. 12 could be recorded from Eri silkworm, while 11 from the root grub. The only difference here was Proteobacteria was more dominant in the gut of Eri silkworm, while Firmicutes were more diverse in the gut of Anomola dimidiata. SEM studies on the gut compartments revealed the presence of chitinous structures in the midgut, anterior hindgut, and fermentation chambers of scarabids. Possibly these structures might be housing the anaerobes. Functional diversity studies were conducted on the cellulolytic gut bacteria, lipolytic gut bacteria, and also their role in nitrogen cycling in scarabids was studied. It was seen that a consortium of anaerobic bacteria were responsible for this nitrogen cycling, and among the 4 identified, Chrysiobacterium was the most dominant one. A study was conducted by Chidananda H.B. on the impact of hot events on biological attributes and antioxidant response in Spodoptera litura under the guidance of Dr. D. Sagar. And this study had 2 objectives, that is to study the lethal and sublethal effect of hot events on different stages of Spodoptera litura, and also the biochemical response of S. litula larvae, which once exposed to the hot The test insect for the study was Spodoptera litura. All the stages of Spodoptera litura were subjected for varying intervals of time to 42 degrees centigrade and 46 degrees centigrade. And after exposure, they were allowed to recover at 30 plus minus 1 degree for 60 minutes. And control insects were maintained at 30 plus minus 1 degree centigrade. And these are the parameters for which observations were recorded. And the effect of exposure duration on egg production was also studied, and it was seen highest egg production was seen in the adults of Spodoptera litura when larval stage was exposed to thermal stress, and no adults emerged or survived after exposure for 20 hours. And same trend was seen for 46 degrees. Activity of various protective enzymes were studied at 42 degrees centigrade and also 46 degrees centigrade. And it was seen increased duration of exposure resulted in higher activity of the protective enzymes. Same trend was seen at 46 degrees centigrade. Another study by Ms. Su, a student from Myanmar, was conducted on the defense response of chickpea genotypes against gram pod borer Helicoverpa armigera under the guidance of Dr. Sagar again. The study had two different objectives and 4 different chickpea test genotypes, and along with one resistant and one susceptible chick were raised in pots, and biological performance and defensive response of Helicoverpa armigera was studied on 21-day-old seedlings. Biochemical studies were performed for 10 different parameters, and it was seen that longer larval duration and lower larval pupil and lowest percentage adult emergence was recorded in test genotypes compared to the susceptible chick, but all these were at par with the resistant chick. The SOD activity, superoxide diamutase activity, was significantly higher in both uninfested and infested plants of the test genotype. Activity of peroxidase increased from 1 to 2.41 fold in all the test genotypes after infestation. Catalase activity, however, showed a declining trend. Polyphenol oxidase also showed increased activity in all the. Heat map analysis was done for the 10 components studied, and it could be inferred that SOD, catalase, polyphenol oxidase, and phenols could be identified as predominant biochemical components for selection of pod borer tolerant varieties. Toxicological studies were done on whitefly Bemisia tabaci populations from different parts of India by Ms. Rajna under the guidance of Dr. G.K. Mahapatra. 3 different objectives were there. She collected 5 populations from Delhi, Punjab, Madhya Pradesh, Haryana, Rajasthan, and compared these populations with the susceptible Delhi population, which was maintained in the lab. Genetic group analysis of all these populations indicated that all of them belong to Asia 2-1 genetic group. Bioassay of Bemisia tabaci populations was done according to IRAC protocols, and it was done for both adult as well as nymphal stages. 5 insecticides were selected for the bioassay. The results are high variation was seen in resistance ratios of different populations. Sri Ganganagar and Bhatinda populations were relatively resistant to cypermethrin, imidacloprid, and thiamethoxam. All the populations were, however, highly susceptible to the new insecticide, diamide insecticide, cyantraniliprole. No cross-resistance was, however, observed between cyantraniliprole and other insecticides. Therefore, this insecticide, cyantraniliprole, can be recommended for insecticide resistance management of Bemisia tabaci in India. Biochemical studies were also done. The activity of 3 detoxifying enzymes, esterase, cytochrome P450 monooxygenase, and glutathione S-transferase were estimated for all the populations, and significant increase in activity of cytochrome oxidase P450 monooxygenase was observed in resistant populations. Molecular studies were also done. The expression of the 3 genes were different for different populations of Biomacia tabaci, although they belong to the same genetic group. Resurgence studies were also done in the field, and Pusa population showed a resurgence after repeated application of cypermethrin at sublethal doses. Another toxicity studies of certain monoterpenoids was conducted against storage pests, Calosobruchus, Cytophilus, and Tribolium, and also enzyme dynamics was studied. It was seen that carvacrol was the most effective monoterpenoid for Calosobruchus, while allyl anisole was effective against Cytophilus oryzae, and thiamol was effective against Tribolium. Biochemical analysis of the effective monoterpenoids was done against the pulse beetle, and it was seen there was increase in the esterase and cytochrome oxidase activity due to carvacrol, and whereas increase in glutathione S-transferase activity occurred due to fenchone, and catalase activity increased due to linalool treatment, thereby showing that different detoxification mechanisms are there against each of these monoterpenoids. 4 different training and webinars were conducted at the Division of Entomology during 2020. The prestigious Pradhan Memorial Lecture was held on Zoom platform. Lecture was given by Dr. Kailash Chandra Zedasai. Coming to the faculty position, we are 28, 17 from the division, other divisions 2, NCIPM 3, ACRIP Honeybees 1, and recently 5 more from Raipur and Jharkhand have been added. Research guides 4 from Delhi, 2 from Raipur and 1 from Jharkhand. 5 more faculty have been inducted during 2020. 1 has superannuated. Number of students on roll was 63, including the 13 outgoing students, and 19 new have recently taken admission. These are some of the awards and recognitions of the faculty. ICAR postdoctoral fellow was hosted by our division. These are some of the international visitors who visited entomology division just before COVID lockdown. About the fellowships, apart from IARI's fellowship, our students are also holding ICAR, JRF, DST Inspire, RGNF, and ICCR fellowships. Employment, as far as student employment is concerned, last year 1 student joined ARS, 2 recently have joined as scientists by ICFRE, and 2 have joined state services. 2 poster awards have been won by students, and publications 15 above 6 and 9 between 5 and 6 have been published. These are some of the student awards. 2 of our students, they have been trained at Israel and USA under NAHEP and CAST last year. 2 of our students, Prashanti and Komal, they have won Best Poster Award and Best Oral Presentations. Apart from academics, our students have also participated and excelled in other extracurricular activities. I acknowledge the help received from Director IARI, Joint Director Research, Dean, Joint Director Education, Joint Director Extension, Head Division of Entomology, PG School, administrative staff, and also our dear students. Thank you.
Thank you, Dr. Devjani. The next presentation is from the Division of Discipline of Nematology. Professor M.R. Khan is making this presentation.
I am presenting significant educational achievement of Division of Nematology during 2020. We have 31 students, 7 MSc, 24 PhD. This year on MSc, 3 PhD students are receiving degree in the 59th IRI Convocation. Broad research themes on which our students worked are insect biocontrol, nematode genomics, nematode biocontrol, and nematode ecology. Santosh Kumar, MSc student, worked on biological activity of a toxin protein TCAV isolated from Photorhabdus heterorhabditis symbiont against insect, with the objective to understand toxicity of TCAV against different genera of insects such as Helicoverpa armigera, Spodoptera fugiparda, Spodoptera litura via in vitro assay. TCAV toxin from Photorhabdus acantha strain of IRI were evaluated by oral feeding and injection to understand immune response, cytotoxicity, mode of actions against 3 insect species. The LD50 values of TCAV toxin of either strain were studied by injecting into insect hemosyl and oral administration. The LD50 values was recorded highest for Helicoverpa armigera, followed by Spodoptera litura, Spodoptera fugiparda, Galleria melonella. Similar trend was observed by oral administration. TCAV toxin was found highly cytotoxic. This caused reduction in number of hemocytes as well as the induction of phenol oxidase mediated immune response. TCAV toxin had caused extensive destruction of midgut epithelial cells, almost similar of BT toxins. Mr. Ashish Kumar Singh studied the draft genome of seed gall nematode Anguina TTC with the objective to sequence genome, assemble, predict, annotate, and compare the genomics with other genomes of plant parasitic nematodes. He sequenced Anguina TTC genome and obtained 164 MB size genome assembly with N50 value of 3000 kb. Approximately 40,000 genes were predicted. Comparative genomics revealed high number of orthogroups among other nematodes. Phylogenetic analysis revealed that Anguina TTC to be closely related to potato dry rot nematode Ditelenchus destructor than pine wilt, root knot, or cyst nematodes. Number of the genes to be associated in the core biological process like developmental, neuropeptide coding, aging related genes, RNA genes, dwarf formation, chemosensory, and sex determination related genes. This genomic information will be helpful in understanding comparative genomics and functional validation studies. Mr. Rami Kasam worked on molecular mechanisms of interaction between Melodogania incognita and foraging biocontrol fungi with the objective to isolate, purify, and morphologically and molecularly characterize of the fungi, then decipher the mechanism of parasitism and to understand the fungi and nematode interaction. He collected 70 soil samples covering 40 plant species from 14 states of India. On 56 pure isolates were obtained and morphologically and molecularly characterized. 17 fungi were identified. All the isolates were evaluated against Melodogania incognita second stage juvenile and found 24 isolates caused 95 to 100% mortality, 40 caused 85 to 94% mortality, 37 caused 75 to 84% mortality, 55 caused less than 74% mortality. Some promising fungal isolate like Bacillomyces tenis, Trichoderma unansi, then Talaromyces spinophilus were identified. Then mechanism of parasitism were further studied. He transformed fungal isolates with showing high mortality against Melodogania incognita under in vitro condition. 3 fungi transformed successfully to express GFP genes and transformed fungi were further confirmed by gene-specific primers. Fluorescent microscopy and same study revealed the mode of parasitism of 3 fungi, Trichoderma, Talaromyces, and Bacillomyces. Transcriptome study was done to understand interaction of Bacillomyces with C. elegans and Melodogania incognita. This fungus secreted huperzine and some chemicals that affected nervous system and nematode locomotion. This was further confirmed by QRT-PCR validation of 2 neurotransmitter genes, AC1 and AC2. Both the genes were downregulated in fungal filtrate treatment and huperzine treatment. Several genes were differentially expressed due to this treatment. Mr. Vivek Narayan Sutar worked on allelopathy effect of host root exudates, host rhizosphere bacteria, and entomopathogenic nematode on penetration and development of Melodogon incognita, with the objective to understand individual and combined allelopathy effect of root exudates, rhizosphere bacteria, and Heterorhabditis indica on the development of Melodogon incognita. He isolated a Melodogon population from infected polyhouse, and then these populations are further multiplied on cowpea and tomato. And the in vitro assay from the root exudates of these two populations of tomato specific J2 and cowpea specific J2 were evaluated. The study showed that tomato specific J2 showed positive attraction to tomato root exudates, but not cowpea, whilst cowpea specific J2 showed a neutral response. Cross-inoculation studies of tomato-specific J2 and cowpea-specific J2 was also studied and found that infection of development of the both populations were significantly reduced on cowpea and tomato respectively. Root exudates were characterized by GCMS. 23 compound was found in tomato, 24 in cowpea, 16 compounds were common to tomato and cowpea, and the uncommon compounds were considered to be important for development of synthetic molecules to disrupt nematode chemotaxis. Rhizosphere bacteria from tomato and cowpea were isolated and characterized. 3 bacteria from tomato, 3 from cowpea were identified. Bacteria isolated from the tomato rhizosphere significantly repelled tomato-specific J2 but not cowpea-specific J2, while cowpea rhizosphere bacteria repelled both the nematode population. All the 6 bacteria were unable to repel Heterorhabditis indica infective juveniles. This study confirms that the allelopathic effect of root exudates on the nematode populations. Rhizosporic bacteria recruited by its host play a significant role in the nematode chemotaxis. The EPN species is unaffected by the root exudates, but it could suppress nematode, indicating its rhizosporic competence. We published 18 research papers. Most of these are 6 to 10 NAS rating. Our PG activities are restricted to online teaching, meetings, examinations, and professor-student interactions. Thank you for your patient listening.
Thank you, Dr. Khan. The next presentation is by Professor V.K. Baranwal from Plant Pathology.
Very good morning. I'm going to present the significant research achievements in the division of plant pathology. The mandates of the division include basic and applied research, to serve as a center for academic excellence, and to provide national leadership. The division has 26 faculty members with head and 1 professor. In this academic session, 4 major fields of study are included in different courses. These include fungal pathology, mycology, virology, and bacteriology. This year we started semester-based program, and in the first semester, 14 courses along with 2 seminar courses have been offered, and in second semester, 12 courses along with 2 courses for seminar will be offered. Currently, there are 78 students, and the faculty-student ratio is 1:3. There are 7 MSc students who are going to receive degree in this convocation, and their names are listed here. Similarly, there are 7 PhD students who are going to receive degree, and their names are listed here. There are 4 theme area of research which students have pursued, and in the theme area 1, that is diagnostics and pathogen characterization, Manjunath Sivappa worked on development and validation of multiplex RT-PCR for simultaneous detection of viruses infecting orchids. There are 4 major viruses such as Medium mosaic virus, odontoglossum ring spot virus, Callanth mind mosaic, and groundnut bud necrosis virus. which he included in his study. And he tested all these viruses through ELISA in orchid samples. He could not detect groundnut bud necrosis virus, and therefore a more sensitive method, that is RT-PCR, was used for detection of all these viruses, including GDMB. He optimized multiplex PCR based on primer concentration and annealing temperature. And the optimized multiplex PCR was evaluated for detection of these 4 viruses in collected samples. He found the presence of these viruses either as an individual infection or in multiple infection. And he could detect ground bud necrosis virus along with other virus in 3 samples, which indicated that multiplex PCR is very sensitive method for detection of either single virus or multiple infection of viruses, including groundnut bud necrosis virus. He demonstrated the presence of infection by 3 viruses in few samples. Sajadun Nabi worked on a virome analysis in mosaic infected cultivars using RNA sequencing. Apple is an important crop of Jammu Kashmir and Himachal Pradesh, but the productivity is very low compared to apple productivity in other parts of the world. So, mosaic disease is an important disease in apple, and it is caused by apple mosaic virus. But the recent studies have indicated that there could be association or of other viruses with mosaic disease in apple. So therefore, he undertook virome analysis in 3 apple cultivar, Red Fuji, Oregon Spur, and Golden Delicious. And he used different softwares for classification and assembly of reads. He found the presence of 5 viruses in asymptomatic as well as in symptomatic plants, along with the presence of 1 viroid, apple hammerhead viroid. He reconstructed the full genome of all these 5 viruses and also developed multiplex PCR for 4 viruses and evaluated it for the presence or absence of these viruses in different apple cultivars. Venkatesh worked on reconfirmation and characterization of pathogenic race of bipolaris medis causing leaf blight of maize in India. And based on the reaction on 4 cytoplasmic male sterile line, he established that bipolaris medis is race O in India. And he also studied on the toxins produced by bipolaris medis. In the theme area, host-pathogen interaction, Ms. Mohli worked on suppressor activity of tomato leaf curl new Delhi virus genes and development of RNAi construct to evaluate their efficacy in inhibiting the virus. So all the suppressor proteins acted on pathogenicity determinant. AB2 has more virulence than others, and transcripts of the suppressor can move systematically. She also worked on study of localization assay through confocal microscopy, and she found that AB2 was localized in host cell membrane, host nucleus, and AC2 was localized in nucleus, and AC4 was localized in host cell membrane. She also demonstrated that AC4 has more suppressing activity than other AB2 or AC2 genes. Aniruddha Chattopadhyay worked on functional analysis of deconstructed genome of cucumber green mosaic virus. He deconstructed by deleting this significant part of replicase enzyme in the genome of cucumber green mottle mosaic virus, and he had 2 deconstructed genome. One was DG1 and the other one was DG2, and he evaluated these 2 deconstructed genes in presence of cucumber green mottle mosaic virus and saw that DG1 could replicate and could move, and also it helped in gene silencing. While DG2 was able to replicate and translate but it was not able to move. This is the first report of development of two novel two component of CGMMB based vector system in India. Achya CK worked on host pathogen interaction between Indian mustard and isolates of Sclerotinia sclerotiorum, and she found that 3 isolates out of 42 isolates were highly virulent, and these virulent strains were isolates were used for screening of resistant and moderately resistant genotypes of mustard. In pathogenomics theme, Miss Manisha Duhan worked on determination of predominant cotton leaf curl virus in northwestern India. In addition to that, she also characterized a beta satellite as well as 2 alpha satellite species associated with cotton leaf curl Multan virus Rajasthan strain. In her study, she found that the isolates were grouped differently than the isolates reported previously, and that only indicates the evolution of new strains of cotton leaf curl virus in cotton cropping system. She also studied the incidence level of cotton leaf curl disease in different districts of Haryana. In theme area 4, plant disease management, Mr. Srinivas worked on determining the resistance of some weed genotypes to stripe and leaf rust pathogen. He characterized 5 yellow rust resistance genes as well as leaf rust resistance genes in exotic wheat material as well as in Indian wheat materials. He also found promising adult plant slow rusting resistance against these two rusts in exotic wheat genotypes. He also found promising adult plant resistance against both types of rust in Indian wheat germplasm. Mr. Akshay Kumar worked on pathogenic and genetic variation among fusarium isolates causing head scab of wheat and identification of resistant sources. 29 fusarium isolates were identified on the basis of morphological, cultural, and molecular approaches, and he identified isolates from Wellington as most pathogenic, and that was used for evaluation of 164 genotypes for fusarium head blight resistance. 4 genotypes showed slow progress of head leaf blight. Harita Mohan worked on identification and validation of germination related genes in teliospore of Telesia indica causing kernel bunt of wheat. She analyzed transcriptomic data of dormant and germinating teliospores, and she submitted the transcriptome data in public domain. She also studied the relative expression analysis of upregulated genes in germinating and dormant spores, and she found that 30 genes were highly expressed in germinating spores but not in dormant spores, while 8 downregulated genes showed upregulation in dormant teliospore, indicating that they may have role in dormancy of spores. Ms. Pooja Kumari worked on characterization of aflatoxigenic Aspergillus flavus associated with maize and aflatoxin resistant gene. She characterized 20 isolates of 5 agroclimatic zones and also found that trypsin inhibitor protein can be used as a biomarker against Aspergillus flavus infection and buildup of aflatoxin in maize kernel. 5 maize inbred lines were found promising against Aspergillus flavus infection and aflatoxin. And the resistance of these 5 lines can be used to develop resistant varieties against this pathogen in breeding. Guru Reddy worked on characterization of pathogen associated with chickpea stunt disease and its epidemiology. Chickpea stunt disease is an emerging problem, and he associated the presence of a new subgroup of phytoplasma in Andhra Pradesh and UP. He developed multiplex PCR for identification of phytoplasma at subgroup level. He also developed duplex PCR for identification of mixed infection of phytoplasma and chickpea chlorotic dwarf virus. He identified for the first time 2 new hosts and 2 new weed species as a host for chickpea chlorotic dwarf virus. In addition to that, he studied on the population dynamics of leafhopper based on temperature index. Avjit Shankar Kashyap worked on induced systemic resistance in capsicum annuum to Ralstonia solanacearum by plant growth promoting rhizobacteria. He found that a combination of Pseudomonas fluorescens and Bacillus subtilis against Ralstonia solanacearum induced better expression of defense-related genes in both root as well as leaf tissues of chili plants in comparison to the control. Mr. Darshan worked on transcriptomic analysis of Chytomium globosum for bioprospection of antagonistic related genes. He used Illumina HiSeq platform, which yielded an average of 20 to 22 million reads with 50 to 58% of GC content. De novo assembly generated about 45,000 transcripts with 28,000 unigenes. The predominant transcripts classified as genes involved in catalytic activity, and 29% of transcripts were involved in metabolic activity. Several mycoparasitism related enzymes, secondary metabolites, and antibiosis related genes were involved in mycoparasitism and antibiosis, and proposed as most effective machinery exhibited by Chytomium globosum. Coming to honors and positions secured by students, many of the students in the division bagged Best Poster awards in national and international conferences. Some of the students also got NAHEP grant for their research work. In addition to that, Mr. Darshan got American Phytopathological Society IPS Award for Plant Health in 2020. Many of the faculty received awards and honors during 2020-21. Dr. Rasmi Agarwal received Punjab Rao Deshmukh Women Scientist Award, ICR. Dr. M.S. Swaharan received K.C. Mehta and Dr. Manoranjan Mitra Award of Indian Phytopathological Society, and he was elected to the Fellowship of National Academy of Agricultural Sciences. In addition to this, many of the scientists like Dr. G.P. Rao, Dr. Robin Gogoi, Dr. T.K. Bagh, Dr. Vikas Mandal, Dinesh Singh, Dr. Kajal Viswas, and Anirban Rai and M.S. Gurjar also received honors and awards from different societies. Many of the scientists participated in the international conferences and got the Best Oral Presentation Award. Some of the students received coveted fellowship like Maulana Azad National Fellowship of Disabled Student, and Prime Minister Fellowship. Short-term training courses and seminars were organized in 2020. The training was organized on preparation and application of microbial formulation of disease management. A Delhi zone of IPS organized virtual symposium on modern trends in systematics and bioprospecting of fungi. In 2021, Students and scientists published a large number of research papers, and 5 of them had NAS rating of more than 10, and these journals were Journal of Pest Science, Frontiers in Microbiology, and Scientific Report. With this, I end my presentation. Thank you.
Thank you, Dr. Baranwal. So we have completed all the 4 presentations of this session. And now I invite Dr. H.B. Singh, the chairperson of this session, for his remarks.
Thank you, madam. Let me congratulate the graduating students and their supervisor first, because they have done very nice work. I'm really overwhelmed to see the presentation done by all the 4 eminent professors. As the previous chairperson mentioned, that the theme of the IRI is to help the farmers. What I have seen all over the presentation, that the basic research has been well connected with the fundamental applied work. What I personally feel, that the work done by students, they are outstanding. In entomology, in agrochemical, in plant pathology, I think the regulatory part is missing. This is my suggestion, that if you connect your, the basic and applied research with the regulatory bodies like Central Insecticide Board, CIBRC, they are the one who regulates the pesticides, the biopesticides, and all. And I remember Dr. Vinita Sen, when she was working in the division of plant pathology, she commercialized one strain known as Kali Sena now. It's available in the market. So instead of telling you one by one detail in the division, what I feel, let me start with the division of entomology first, and agrochemicals, then finally this. So in agrochemicals, Dr. Neera Singh made a very good presentation on development of agrochemicals and formulations. You see, these formulations are very much needed by the agrochemical industries. So if we connect the students of PhD student work, MS student with the requirement of the industry, that will be a very novel work. I'm extremely happy to see the type of publication in a very high impact journal, particularly in the agrochemical division. They have published very good papers. The faculty got large number of awards. 5 ARS students have been produced. So overall, I feel they have done outstanding work. Similarly, in entomology, excellent work has been done on insect biosystematics. See, very few institutes in our country, they are working on this particular area. So I think this is a notable contribution by the division of entomology on the insect biosystematics. Then they reported 2 new genera of leafhoppers and many new species. This is again a very outstanding work, and molecular phylogeny was carried out by the division. So this is again a very, very important work done by the students of entomology division. In nematology division, nematode genomics, I think the only institute working in the country is IRI. They have done good work, and the genome sequence is available in public domain. That is very good thing. At the last, in the revision of plant pathology, excellent work has been done on viruses and phytoplasma diseases. Virome analysis of apple mosaic virus is again a very, very important work done by IRI. So, I congratulate the supervisors and the student both for doing this excellent job. the work on pathogenomics, the plant disease management, particularly the biological control using, the kernel bunch of wheat and all. This all really has been done by the students and the supervisor in a very nice way. So my overall assessment is the work done by all the 4 divisions are outstanding. As I said, we have to connect with the regulatory body so that the results can go to the industry and ultimately to the farmers. This is what exactly we want. That is all I wanted to say, Dr. Asmi. Thank you so much.
Thank you, sir. Very well taken, sir. Because we know that we are producing products in Division of Agriculture, chemicals and many biocontrol agents, and their formulations have been developed in pathology also. And also in entomology, we have many parasitoids identified for insect pest management also. So we have some linkages we are trying to develop, and we are trying to get some of the products commercialized through our ZTM unit also. So we are really into it, sir, and definitely your support and guidance we will be requiring in between also. We will be in touch with you. And very good remarks you have given, sir. I agree that it's a lot of basic work is being done, and that has to be developed into a product. That is definitely is our next goal, sir, for the School of Crop Protection. Yes.
Madam Rashmi. Madam Rashmi, can
Sir. Sir, want to say something, sir?
Yeah, I just...
Yes.
Yeah, let her finish, sir.
Yes. Anupma.
Yeah. So, namaskar, sir. Myself, Dr. Anupma, Head Agricultural Chemicals Division. Thank you very much for your encouraging words. Sir, regarding the inventions and the work taken and reported by the students of agricultural chemicals, sir, we are very well now in collaboration, and this is going to be fostered in several formal platforms with the agrochemical industry. Just yesterday, we had a meeting with the Dhanuka Group MD and his team on the products and that are there in our basket. And sir, I'm assuring through this platform that all the problems that are being taken now, they are taken after due consultation with the industry based on their surveys at the ground. And sir, with a constant time frame, all the specifications of the formulations, they are being followed as per the CPAC and the CIBRC, that is regulatory body requirements. So that the data which is generated here, that is for the industry acceptance, and then the registration of that can be taken up at the very fast. So this is how, sir, we are doing. And we have now submitted a flagship program also through division, through the institute, and that will be the, sir, platform for the public-private partnership. And so sir, we assure you that the products will be there from the students as well as the faculty outcome in very near future. Thank you very much, sir.
Thank you, ma'am. Another suggestion I am to make, see, as I said, you have very good publication. Faculty has been honored for that. But I didn't see any presentation having the portfolio of patents. I don't know whether the patents have been filed or not. Yes.
Yeah. Sir, Division of Agricultural Chemicals has in past 4 years been granted with more than 10 patents in various inventions, and they are both national and international patents. And the work of Ms. Tracy Bishwas, which was presented today by Madam Neera, that was on the combo formulation of CCC and tebuconazole. The provisional patent of that, the filing process, it has been initiated, and it has been granted approval by the ITMC also for the patent filing. So, sir, it is in process, and this year we'll see, I believe that this year we'll see number of more patents on that. But sir, the quality of the inventions are more important, so that we are, very careful about filing the patents before really getting reproducible results. So, sir, we are onto it, sir, so that our IP is duly protected. Yes, sir.
Yes, sir. Sir, I will supplement here. from the Division of Plant Pathology also, sir, some documents have gone to ZTM. So we did not present here, but definitely we are into it. And since it is partly covering only few students' research work, so it is not covering the whole of research activities being carried in the different disciplines. So we are not covering the whole gamut of research activities. So we did not include some other activities which are being taken at the division level. So definitely your points are well taken, and we will be doing it further on it. Yes, Seema. Dr. Seema, please.
Yes, ma'am. Yes, ma'am. So we have come to the end of this session. So I would like to thank Dr. H.B. Singh, sir, for chairing this session and also for providing the valuable suggestions, remarks, and critical comments on all the four disciplines which were presented here. Thank you so much, sir. Thanks for all the appreciation. Thank you. Thank you
Sir,
so
you
much.
can.
And I would also like to thank all the four speakers who made excellent presentation in this session, and all those who have joined this session. Thank you so much. Thank you.
Sir, if you have time, sir, you can keep joining, sir, all the sessions. Yes, sir. We welcome you, sir. Okay.
Continue.
Yes, we will continue. Pre-lunch session will continue.
Yes.
So yeah, I have pleasure and an honor to welcome our chairperson of the next session, Dr. Anil Kumar Singh. Sir, we welcome you, sir, on this session on natural resource management. So this is the largest school having 7 disciplines, sir. So we had presentations of crop improvement in the morning, and just we finished the School of Crop Protection presentations. So now we will have the School of Natural Resource Management presentation, sir. So on behalf of Director IRI, on the behalf of faculty, and on my own behalf, sir, I profusely welcome you, and looking forward to your suggestions and observations on the presentations made by the different professors on the School of Natural Resource Management. Sir, the professors will be giving the presentations, and there are total 70 theses, sir. The work of which will be presented by the professors of various disciplines, and which includes 42 MSc and 28 PhD theses. And again, I would like to mention here, sir, this is not the total research activities and which are being carried out under the School of Natural Resource Management. It's only that all the students' work is included who are getting degree during this 59th convocation. And this is the third day of our 59th convocation week program. And main convocation we are having on 12th of February. So, sir, with these brief introductions, sir, Dr. A.K. Singh was a former Vice Chancellor, Rajmata Vijay Raje Scindia, Krishi Vishwavidyalaya, Gwalior. And also he was former DDG, NRM, ICR. So, sir, we welcome you to chair this session. And I would like to request our director, Dr. A.K. Singh, to formally introduce the chairman to the all invitees.
Thank you, Dr. Rasmi, and what a pleasure for A.K. Singh to welcome Dr. A.K. Singh. Welcome, some hearty welcome, sir. And Dr. A.K. Singh, as you all know, he has been associated with IRI for very long. He did his master's and PhD from IRI, and then he had several postdoc and overseas exposure at University of Monheim, Germany, and then at University of Arizona, International Rice Research Institute, full postdoc. And he has made tremendous contribution to teaching and research in field of soil, water, nutrient, plant relationship, remote sensing application, and use of modern tools for water management. Dr. Singh is awarded with Dr. Sri Subramaniam Outstanding Teacher Award, which is a rare distinction for any teacher, and also IRI Best Teacher Award. He is Fellow of National Academy of Agricultural Sciences, and in his administrative capacity, he served as Project Director, Water Technology Center, Deputy Director General Crop Science, and Vice Chancellor of Rajmata Vijay Raje Scindia Agriculture University at Gwalior. It is our pleasure to welcome you, sir, and we look forward for your constructive suggestions for improving our program. Thank you very much.
Dr. Seema Jaggi.
Yes,
Please
I would
take it.
like to invite Dr. A.K. Singh for his initial remarks.
First of all, thank you, Dr. Ashok Kumar Singh, for giving me this opportunity to be part of the convocation celebrations of IRI. As you all know that I have been at IRI for a very long time, and it's always a pleasure to be there and listening to the work that is being done in IRI. I think I will, and certainly thank you very much for a very warm welcome. Whatever I have achieved in my professional career is because of my training at IRI all through. So that way I'm very grateful to IRI for what it has given me. And I think I will listen to the presentations and then probably give my comments. Natural resource management is one of the, I consider the important aspect of agriculture research and education, because it is going to play a very critical role in enhancing productivity and also sustaining it in the future. And so I believe these 7 department divisions which will be presenting their work will ultimately lead to improvement in all input use efficiency at various levels in the crop production system. So with these words, once again, thank you very much for inviting me, and I think we can go ahead with the presentations so that I'll give my comments at the end. Thank you very much.
Thank you, sir. Thank you, sir. So we have 7 presentations in this session: agriculture engineering, agriculture physics, agronomy, environmental sciences, microbiology, soil science and agriculture chemistry, and water science and technology. So we start with the agriculture engineering. The presentation is by Professor D.K. Singh.
Honorable Chairman of the session, Director IARI, Dean and Joint Director Education, Convener of the session, faculty members and students. Welcome to the presentation of Division of Agricultural Engineering. Students' research in the division is carried out in 3 subdisciplines, namely farm power and equipment, soil and water conservation engineering, and agricultural structures and process engineering. We have 75 faculty members, that includes the faculty members at CII Bhopal. As of now, we have 126 students on roll. 13 students will be receiving degree this year. Externally funded research projects have been great support of our students. As of now, we have 14 externally funded projects in our division. Some important research projects are paddy straw residue management through in situ microbial decomposition with mechanical intervention, off-grid clean energy cooling for affordable storage of perishable product for BOP farmers, customization of appropriate technologies and practices for eco-friendly and economical management of crop residue, Design Innovation Center under Spoke and Hub model of Ministry of Human Resource Development, Farm Machinery Testing Facility of Government of India, development of solar-powered variable swath herbicide applicator, robot for high-value vegetable crops, development of smart mobile e-power unit with machine implementation for small farmers, development of sensor-based precision seeding profit module for cultivators. So we can see that we have projects from development of small machine to the application of IoT and drone and robotics. This year, students' research is being presented under 6 themes, namely resource conservation technologies, precision farm equipment, integrated water resource management, precision irrigation and crop productivity, solar-powered farm equipment and storage structures, and post-harvest technologies for quality detection and storage. There was one thesis under the theme resource conservation technology. As we know, the continuous use of rotavator creates a hard pan in the subsoil and creates an unfavorable condition for crop growth and affects the yield. A study was carried out by a PhD student at Farmers Field in the Samli district of Uttar Pradesh to investigate the extent of subsoil compaction due to continuous rototilling and determine the effects of subsoil compaction on crop response for suggesting mitigation strategies. 5 tillage treatments, namely conventional tillage, zero tillage, rotary tillage for less than 4 years, rotary tillage for 4 to 8 years, and rotary tillage for more than 8 years were considered by her. She reported a severe compaction at the depth of 37 centimeter in case of rototilling for more than 8 years. Yield of rice and wheat were lowest in the field under rototilling condition. Based on the study, it was concluded that frequent application of rotavator for longer period creates compact layer below the tilling depth. Therefore, subsoiling at 1 to 1.5 meter once in 3 years can be option for managing soil compaction. There were 3 theses under the precision farm equipment theme. A sensor-based liquid fertilizer metering system was developed to facilitate the controlled and precise application of required amount of fertilizer by a MTech student. Device was validated for UAN, urea, and aqueous neem-coated urea fertilizers. Results showed a close agreement between measured values and values measured by the device. Another MTech student developed a steering control system for Mahindra and Yuvraj models of the tractor. Device was mounted on these tractors and its performance was evaluated in the field. Maximum lateral and front wheel angle deviations with this device at 2.8 km per hour speeds were much lesser than the manual guidance system. A PhD student at CIE developed a tractor-drawn pneumatic planter for rice and wheat. Planter consists of a drum-type metering device with 10 orifices and uses petrol engine-operated aspirator. Planter was evaluated in the field condition. Field capacity of the planter was 0.42 hectare per hour and 0.44 hectare per hour for rice and wheat respectively. Cost of machine is about 92,500 rupees with a payback period of 7.7 years. There were 2 theses under the theme Integrated Water Resource Management. A MTech student conducted a study at farmers' field in Sundarbans Delta to evaluate the recharge characteristics, determine the spatial and temporal variation in the soil and groundwater salinity, and develop water utilization plan. The study area is prone to the backwater flow from Bay of Bengal, cyclone, and seawater intrusion. Soil and groundwater salinity are the limiting factors for agricultural production in the area. Observation wells were installed in the area for monitoring groundwater levels and quality. Hydrus 1D and water balance models were used to assess the groundwater recharge from the paddy field and ponds. The results showed that the area is not favorable for groundwater recharge because of low infiltration rate and high silt and clay content in the soil. Pond in the area can be used for rainwater harvesting and storage, not for groundwater recharge. Soil and groundwater salinity in pre-monsoon period was higher than the post-monsoon period. Irrigation intensity with the water stored in the pond could be increased by 53% in an area of 0.2 hectare if farmer cultivate tomato on entire isle area and rabi paddy in half of the area of the mainland. Difference in water levels between observation wells installed adjacent to the pond and 33 meters away from the pond was not much. This also suggests that the pond cannot be used for groundwater recharge. Irrigation application efficiency is a major concern in our country. A MTech student studied the performance of a miner under Eastern Zone High-Level Canal Command in Bihar. He used tools such as remote sensing, GIS, and crop water model in his study. He found that the irrigation application efficiency in head, middle, and tail reaches were 51%, 53%, and 59%, and canal releases during initial stages were more than the required, and during peak demand, inadequate water was released in the canal. Our students have been working on various aspects of water management for greenhouse cultivation. A MTech student used computational fluid dynamics for irrigation scheduling in greenhouse. He developed a CFD tool for simulating evaporation from greenhouse and evaluated under field condition. Simulation results were in close agreement with the observed values and can be used for greenhouse irrigation scheduling. Hydroponics are becoming very popular nowadays. MTech students designed and developed a bamboo-based automatic hydroponics structure and evaluated its performance with the mint crop. Different components of the system are shown in the figure. Developed hydroponic system was found to be successfully controlling various parameters of nutrient solution. There were 2 theses under the theme solar-powered farm equipment and storage structure. A student designed and developed a solar pedal operated paddy thresher. Developed thresher was evaluated with PB 1509 variety of the rice. Evaluation results showed that the solar operated paddy thresher had higher threshing efficiency and better quality of threshed paddy grains. Under the same theme, a student developed a ANN model for simulating the quality of stored horticultural produce. Model was used to simulate the quality under various types of storage structure. The simulation results showed that the green tomato can be stored up to 33 days in solar refrigerated evaporative cooling structure, whereas spinach can be stored for 13 days. There were 3 theses under the theme Post-Harvest Technology for Quality Detection and Storage. A PhD student at CII used PIS NIR hyperspectral imaging protocol for estimation of nitrate in harvested leafy vegetable and developed a model for assessing nitrate content. Based on the evaluation and validation results, he concluded that the model can be used to assess the nitrate content in leafy vegetable and can be adopted by the food industries for grading. He also found that increase in nitrogen fertilizer dose led to deterioration in quality of the leafy vegetable. Another student at CIA Bhopal conducted a study to find out the efficacy of ozonation to disinfect the stored chickpea grain. He fabricated ozonation structure which was equipped with the ozone sensor and alert system. He observed that adult and egg mortality was 100% and 1000 ppm and insect egg cell collapse at 700 ppm. Dehulling efficiency increased at 750 ppm and cooking time decreased at 1000 ppm. A PhD student conducted a study on use of ultrafiltration techniques for preparation of soya protein with improved functional property. Towards the end of my presentation, I would like to inform you that the division has organized 2 workshops and added several equipments in the newly created food testing and analysis laboratory. We have added integrated projection system and digital boardroom in the class. Our students have published 10 research papers. 4 of them they are published in NAS rating more than 6. Our students are well placed. 7 of them they have joined the PhD. And faculty members have received several honors and recognition. At the end, I thank my students, head of the divisions and faculty member, Dean and Joint Director Education and Director IARI for providing necessary support. Thank you.
Thank you. Thank you, Dr. Singh. Thank you. The next presentation is by Professor Vinay Kumar Sehgal from the discipline of Agricultural Physics.
Respected chairman, dignitaries, scientist colleagues, staff, students, ladies and gentlemen, I am going to present the significant educational achievements in the discipline of agricultural physics during the last 1 year. Agricultural physics is a unique multidisciplinary subject in which research, teaching and training is conducted in one or more of its subdisciplines of agriculture meteorology, plant biophysics, remote sensing, GIS, soil physics, and lately also started in sensors and instrumentation. The subdisciplines make use of modeling, data analytic techniques to solve real-world problems ranging from field to region. This year, 6 students are graduating in the discipline of agricultural physics, 2 PhD students and 4 MSc students. Their research work is categorized under the themes of remote sensing and crop modeling, soil physics and conservation agriculture, and agrometeorology and machine learning. In the area of remote sensing, the PhD student Rajkumar Dhakar worked on regional wheat yield prediction using remote sensing inputs with dynamic crop weather models and having 3 objectives. First one was to evaluate simulation model performance at research farm scale and farmer's field. Second was to retrieve crop parameters from satellite images and then assimilate into crop model to compensate for some of the errors in input which may not be available for all the fields in a region. And third one is to develop a prototype toolkit to forecast the crop yield at a finer scale using in-season remote sensing data
and weather forecast coming from a weather prediction model. He conducted experiments in IRI farm and farmers field in Mumtazpur and Lokra villages of Gurgaon. Three types of models were used by him. First one was the canopy radiative transfer model to estimate crop biophysical parameters like leaf area index, chlorophyll content from remote sensing images. A modified infocrop model which can assimilate remote sensing derived crop parameters and correct itself based on in-season information, and a weather forecasting model which provides the weather forecast at a finer scale. The whole methodology has been converted into a Python software with a user-friendly interface. These are the grain yield maps which are generated by assimilating in-season leaf area index into crop model successively during the crop season, and these maps shows that a large variability in crop yield at farmer's field scale could be generated. And if we see how the errors moved as we keep on assimilating more and more number of remote sensing derived leaf area, the errors in decrease from 20% to something less than 10%, around 7 to 8%. Whether we use actual weather or we use a forecasted weather which has been bias corrected using adaptive bias correction technique. The whole prototype was converted into a Python toolkit software with a user-friendly interface and is available on IRI website for download and used by different stakeholders. In the subject area of soil physics and conservation agriculture, PhD student Brijesh Yadav studied the effect of conservation agriculture on soil hydrothermal processes and nitrogen dynamics in wheat and mustard using two different models, Hydrus 2D and EPSIM. He took observations in long-term conservation agriculture experiments in IRI under two cropping systems, that is maize wheat mung wheat and maize mustard mung wheat. The broad two contrasting treatments were zero tillage with residue and conventional tillage with residue. The table shows validation of Hydrus 2D model for wheat to simulate depth-wise soil water, soil temperature, soil nitrogen under different ZT and CT treatments. It showed that the model performance was good in terms of high R-square and high D-index value for wheat, and similar results were obtained for mustard also. The study clearly concluded that zero till with residue resulted in better soil physical conditions over conventional tillage with residue, like decrease in bulk density, increase in saturated hydraulic conductivity, as well as increase in field capacity significantly under both cropping systems. There is the zero till with residue also resulted in moderation of temperature in the soil, and there was higher root water uptake. This all resulted in better grain yield in wheat, which increased by 18 to 30%, and mustard yield increased by 10 to 18% under zero tillage with residue as against conventional tillage with residue. This study also showed that Hydrus 2D was performing better than EPSIM model in simulating soil water content, soil temperature and soil nitrate. And the long-term simulations over 30 to 40 years for Northwest India showed that with zero till plus residue could result in increasing water productivity by 19.2% and nitrogen use efficiency by 9% in wheat. Mr. Trideep Ghosh, MSc student, also worked in the area of soil physics and conservation agriculture by employing a novel technique of X-ray computed tomography imaging to quantify porosity and pore size distribution in soil and how it affects the carbon mineralization under conventional and conservation tillage practices. He took observations in IRI long-term field experiment at the time of sowing and at harvesting of maize crop from different depths under conventional tillage and zero tillage treatments like permanent narrow bed with residue, without residue, permanent broad bed with residue and without residue, and flat bed with residue and without residue. These are 3D views of soil cores generated using multi-angle observations by X-ray computed tomography. The images show higher macro porosity and lower microporosity due to soil disturbance in conventional tillage. It shows uniform distribution of soil pores and better pore connectivity in permanent broadbed with residue, and it showed compaction in permanent flatbed system without residue. The study clearly showed that X-ray computed tomography can be used for characterization of pore size distribution in soil, and the study concluded that with the incorporation of residue, there was 5% more total porosity as compared to non-residue applied pots, and the highest hydraulic conductivity infiltration rates were recorded under permanent broadbed with residue, which showed that long-term adoption of conservation agriculture practices improves soil structure and pore continuity. And among different pore classes, the pore diameter of 0.11 to 0.5 millimeter had positive correlation with carbon mineralization. Sonia Dagar, MSc student, studied the stability and mechanical properties of soil aggregates under tillage treatments of conventional and no tillage, with and without residue, and at different levels of nitrogen application. The observations were taken in maize wheat long-term experiment conducted in IRI. She concluded that no tillage and crop residue mulching increased percentage of water stable aggregates as well as their mean weight diameter. The carbon and nitrogen concentration in bulk soil as well as in macro aggregates increased under no tillage with crop residue mulching. The carbon and nitrogen sequestration potential was also higher under no tillage with crop residue, and it increased with increasing dose of nitrogen. The no tillage and crop residue also resulted in very high labile carbon and labile carbon pools than conventional tillage. The activation energy and temperature sensitivity of soil carbon mineralization under no tillage and crop residue mulching was much higher than conventional tillage with no mulching. So it overall shows the benefits of no tillage with residue mulching. Nandita Mandal, MSc student, undertook a study on characterization of rice blast disease using hyperspectral remote sensing. This was a new initiative in which blast disease score was formulated on a scale of 0 to 9, and she took their hyperspectral measurements in rice crop grown in the Hawalbagh station of Vipcas, Almora. The study identified two wavelengths at 1148 and 1301 nanometers, which had highest sensitivity to blast disease. Using these wavelengths, she proposed two new vegetation indices, normalized difference blast index and ratio blast index, to estimate blast disease score. Besides, she explored different machine learning algorithms to predict the blast disease severity using hyperspectral reflectance. The multivariate adaptive regression spline based model performed best among the 4 machine learning algorithms. In the subject area of agrometeorology, Shreya Gupta, MSc student, explored different machine learning algorithms to predict wheat yield in northwest India at the end of season, as well as to make multiple forecasts at different growth stages during the crop season using weather variables. The machine learning models were developed and tested in R computer language. The study showed that for all the stations, the LASSO, that is least absolute shrinkage, and selection operator model performed best with the predictions error less than 5% at the end of the season, and errors were less than 10% when used in forecast mode at different growth stages. Happy to share that in the last year, 29 peer-reviewed publications came out of students' research. 4 publications were having NAS rating 10 and above, 8 between 8 and 10, 1 between 7 and 8, 12 between 6 and 7, and only 4 papers were there whose rating was below 6. Besides, our students bagged some awards also, and in the department we received 2 postdoctoral fellows, one SERB National Postdoctoral Fellow and another ICR Postdoctoral Fellow. Regarding placements, 2 of our students were selected in ARS. One of our earlier student got selected as Scientist D in India Meteorological Department, and out of 4 MSc passed out students, 3 were selected in IRI to further pursue PhD. Our faculty conducted 9 online trainings and meetings during the year. The 2 well-attended and well-received webinars were conducted by Dr. Sahu, one on sensor and sensing in precision agriculture, under Vaibhav Summit and one on drone remote sensing. Dr. Rajkumar conducted 7 distance learning trainings in collaboration with ISRO. It is a matter of pride that our faculty, Dr. P. Krishnan, received NAS Fellowship. Dr. Ananta and Dr. Monica also received different awards. Besides, our faculty has been contributing to various national level and ICR programs like developing guidelines for drought monitoring and management and the revision, use of technology for crop yield estimation under Pradhan Mantri Fasal Bima Yojana, developing BIS standards for automatic weather stations, etc. With this, I thank you all for the patient hearing and acknowledge the support and contributions from our students, faculty, head of department, Dean and Joint Director Education, Director Sir, and postgraduate school staff. Thank you.
Thank you. Thank you, Dr. Sehgal. We now have the presentation from the agronomy discipline by Professor T.K. Das.
Respected and Honorable Chairman of this session, respected Honorable Director IRI, Dean and Joint Director Education, Convener of this session, heads and professors, distinguished scientists, students, ladies and gentlemen. I welcome you all. Myself Dr. T.K. Das, Professor Division of Agronomy, presenting the significant educational achievement of the Division of Agronomy during 2020-21. Agronomy is one of the leading divisions of IRI. The teaching program in agronomy was started way back in 1945 and IRI associateship equivalent to MSc degree was given first in 1947. MSc was started in 1958. Till date, 258 associateship, 336 MSc and 529 PhD degree have been awarded. Presently, student-faculty ratio in the division is 3:1 and student guide ratio is 4:1. There are 30 faculty members presently in the division. 30 students, 15 MSc and 15 PhD got admitted during 2020-21. They belong to IRI New Delhi, IRI Jharkhand, IRI Assam and NIBSM Raipur. 15 students, 10 MSc and 5 PhD are receiving degree in this convocation. Smita Tripathi, under the chairmanship of Dr. S.L. Meena, studied on integrated phosphorus management for upland directed rice in northeastern hill region, Barapani, Meghalaya. Northeastern hill region has almost 80% soil under soil acidity and P availability in soil is very low, almost 6 to 8 kg per hectare. She tried solely organic, solely inorganic, organic plus inorganic sources of P in the treatment. She studied different pools of phosphorus. The aluminum bound P pool was higher than iron bound P at pH 4.2 and total inorganic P was higher than total organic P due to dominance of mineral bound P, and then soluble as well as loosely bound P, which is directly related to crop uptake, was significantly higher in the organic based treatment, that was 25% FYM, 25% vermicompost and 25% poultry manure plus PSB. The microbial activity such as microbial biomass carbon, dehydrogenation activity, acid phosphatase activity were significantly higher in this organic treatment. This inorganic treatment gave 8% higher grain yield than RDP, 5, 10 and 17% increase in hulling, milling and head rise recovery over control, 8 and 5% increase in gross and rate return than RDP, and 10% increase in phosphorus uptake by grain than in organic treatment. Osolla HM, under the chairmanship of Dr. Y.B. Singh, studied on phosphorus management in direct seeded rice and black gram intercropping system under rain-fed upland soil in Hazaribagh, Jharkhand. She tested 4 cropping system with 3 P management options, and it was found that rice equivalent yield and rate return was similar but higher in Sol background and 4:1 and 4:2 Rosen ratio of rice and black gram and 50% RDP plus PSB plus MF treatment. It was also found that dehydrogenase activity, acid phosphatase activities and microbial biomass carbon was higher in the Sol background as well as 50% RDP plus PSB plus MF treatment. Rajkamal Meena, under the chairmanship of Dr. Dinesh Kumar, studied on foliar fertilization of potassium thiosulfate on agronomic performance of potato. That there is ICR recommendation for potato, that is 140 kg nitrogen, 75 kg P2O5, 110 kg K2O, 5 kg zinc and 30 kg sulfur. We know that potassium greatly influences starch synthesis and potato tuber. Potassium thiosulfate through foliar feeding of potassium can enhance nutrient use efficiency by supplying K and sulfur. Potassium thiosulfate was sprayed 9 liter per hectare at 35 and 55 days after planting of potato, and it was found that total tuber yield, cheap grade tuber yield as well as net return and net BC were highest under the ICR recommendation plus 100% potassium thiosulfate treatment, and also higher uptake of N, P, K and sulfur was achieved in this treatment. Venugopal Reddy S, under the chairmanship of Dr. S.S. Rathore, worked on green manuring for nutrient management in rice under northeastern hill region, Barapani, Meghalaya. He tried green manuring of buckwheat and rice bean with 100% RDF, and it was found that buckwheat green manuring and rice bean green manuring 100% RDF gave higher grain and straw yield net return as well as profitability in DSR. Protein content as well as protein yield was also significantly higher in this green manuring treatment with 100% RDF. It was also found that higher values of SOC, available NPK and microbial activities such as dehydrogenase, acid phosphatase and microbial biomass carbon was higher in these two green manuring treatment with 100% RDF. Mutiranjan Pradhan worked on precision nutrient and weed management in direct seeded upland rice under Eastern Plateau and Hill region, Jharkhand. Under the chairmanship of Dr. S.S. Rathore, he had 3 precision nutrient management combined with 4 weed management treatment, and weed control efficiency and rice grain yield was higher under nutrient expert guided NPK application and weed free treatment. However, LCC based nitrogen application plus full dose of P and K was comparable with nutrient expert based nitrogen application, and pendimethalin followed by hand weeding treatment was comparable with weed free. Yard number, leaf area, protein content and protein yield are significantly higher under nutrient expert guided NPK application and weed free control treatment, but pendimethalin followed by hand weeding treatment was almost comparable with weed free control treatment. Munni Chinio, under the chairmanship of Dr. Raj Singh, studied on weed management in rain-fed palm millet under conservation agriculture. She had 3 tillage treatment combined with 3 weed management treatment, and it was found that grain yield and weed control efficiency and net return were higher under 0 tillage plus residue treatment than in CT or ZT alone treatment. Atrazine followed by 2,4-D treatment gave higher values almost of all these parameters. 0 tillage plus residue had higher water use efficiency, but 0 tillage had higher energy productivity use efficiency because of residue has energy value, and atrazine followed by 2,4-D treatment gave significantly higher water use efficiency as well as energy use efficiency and productivity. Sachin Singh, under the chairmanship of Dr. Amal Ghosh, studied on nitrogen and weed management in direct seeded rice for higher productivity and profitability in eastern India. He studied for weed management treatment combined with 3 nitrogen management treatment, and it was found that rice yield was similar under 50% FIM plus real-time nitrogen management using LCC and RDF, and was significantly higher than in others. Weed free control, however, gave highest yield, but brown manuring and pendimethalin followed by bispirobate treatment were on par. Introduction of 50% FIM plus RTNM or RDF with weed-free control and brown manuring gave similar yield. He studied several microbiological parameters: urease activity, dehydrogenase activity, microbial biomass carbon, as well as available soil nitrogen are higher under the 50% FIM and real-time nitrogen management using LCC base as well as in brown manuring treatment. Sandeep Kumar, under the chairmanship of Dr. B.K. Singh, studied nitrogen and water management of maize grown under conservation agriculture. He had 3 sets of treatment like crop establishment, nitrogen scheduling, as well as irrigation regime. And it was found that CA gave 15% higher yield than CT, 75% nitrogen applied basal, and then green seeker added nitrogen applications gave higher maize yield, and combination of CA or 25% available soil moisture deficit with 75% or 50% basal nitrogen plus green seeker added nitrogen application treatment were similar but gave higher yield than in others. It was observed that net monetary gain due to CA was 29,436, due to 25% available soil moisture deficit was 13,516, and due to 70% nitrogen application basal plus green seeker 11,067, and total there was a 54,019 rupees net gain was achieved due to adoption of this technique. Vishwajit Rana, under the chairmanship of Dr. C.M. Porihar, studied optimization of water and nitrogen application through subsurface drip irrigation in rice under conservation agriculture based rice food system. He had 3 sets of treatment: transplanted rice as well as direct seed rice, and levels of nitrogen, and subsurface drip as well as flood irrigation treatment. And it was found that zero till DSR with subsurface drip irrigation and 100% nitrogen application had 9% lower rice yield but 76% higher irrigation water productivity and 48% irrigation water saving than transplanted rice. Flag leaf nitrogen as well as specific leaf nitrogen were significantly higher in this zero till DSR, as well as transplanted rice treatment. About 39% and 82% water lost through ET in phase 1 and phase 2. And this need 5 times more irrigation water here than subsurface drip treatment. During phase 2, deep drainage was 83% lower from subsurface drip than PTR and had 77% less water use. Adoption of SSD treatment over PTR could save 80% and 77% of applied water. Agronomy Achievements, the chairmanship of Dr. C.M. Porihar, studied on optimization of rate and time of nitrogen application through subsurface drip fertigation in maize under conservation agriculture based maize wheat system. He had 3 sets of treatment: CA, that is permanent bed with and without residue, and then flood and subsurface drip irrigation treatment, and levels of nitrogen. And it was found that CA based permanent bed with residue with subsurface drip irrigation and 150 kg nitrogen application gave 20% higher maize yield than furrow flooded plot, and water SSD and furrow irrigated plot were 628 and 728 millimeter. It was observed that photosynthetic nitrogen use efficiency, irrigation water use efficiency and productivity, and radiation use efficiency were significantly higher in CA based permanent bed with residue, subsurface drip irrigation and 150 kg nitrogen treatment. Subsurface drip system gave overall 42% saving in irrigation water. Subsurface drip gave 21% higher yield of maize with 11% lower water input that will reduce overdrafting of groundwater. And this treatment also led to 28% higher agronomic efficiency of applied nitrogen. Gaurav Ghosh, under my chairmanship, studied on impact of conservation agriculture on crop productivity, soil properties and microclimate in rice food system under a long-term conservation agriculture. He studied in the 6th and 7th year of the long-term conservation agriculture experiment, and there are double zero till, triple zero till system with and without residue compared with CT system, and it was found that rice leaf area was continuously higher throughout the crop growth stages in TPR compared to CA system, whereas reverse was true for wheat.
lunch time पे
Here, triple zero system with
इसको अंदर
residue
plug in किसी
had
किसी में
11%
ऊपर
lower
भी डाल
rice yield but 13% higher wheat yield and 34% higher system productivity. And this system also gave comparable net return from rice, but almost
सर्टिफिकेट लेके आएंगे
17,000
जो बाहर से आ रहे हैं
higher net return from wheat and rupees
ये तो कुछ हो ही
86,000
नहीं दे रहा
higher
है ठीक
net return over transplanted rice followed by conventional till wheat system. The triple zero system with residue gave 50% higher system water productivity than CT system. However, its energy use efficiency productivity was lower than without residue treatment, and it significantly improved soil carbon as well as aggregation in terms of water stable aggregate and mean weight diameter significantly. Mohammad Hasnain, under the chairmanship of Dr. B.K. Singh, worked on optimization of fertilizer input under varying tillage and residue management option in maize wheat system. He had 4 crop establishment treatment, CA with and without residue and permanent raised bed with and without residue, combined with soil test bed nitrogen application, nutrient expert and nutrient X plus green seeker added nitrogen application. It was found that system productivity and net return were higher under this permanent raised bed with residue and nutrient X plus green seeker added nitrogen application. PRB plus R and nutrient expert plus green seeker led to lower bark density and higher microbial biomass carbon and dehydrogenase activity and partial factor productivity of nitrogen, phosphorus and potassium in maize compared to other treatments. Mohammad Hasnain also studied nitrous oxide and carbon dioxide emissions in maize and wheat, and he found that nitrous oxide and carbon dioxide emissions, global warming potential were higher, but GHG intensity was lower in CT plus residue treatment than in permanent raised bed plus residue treatment, and CT or permanent raised bed with residue had higher emission than without residue, and nutrient expert plus green seeker added nutrient application had lower emission than soil test based nutrient application. Haris Amen, under the chairmanship of Dr. A.K. Chaudhary, studied on phosphorus management in conservation agriculture based maize wheat system. He had 3 crop establishment treatment combined with 3 phosphorus fertilizations, and it was found that photosynthetic rates in maize and wheat were significantly higher in permanent raised bed with residue and 50% P plus PSB plus MF plus 2 foliar sprays of DAP at the rate of 2%. It was found that system productivity and net return were higher in permanent raised bed plus residue treatment and 50% P plus PSB plus MF plus 2 foliar spray of DAP treatment. However, energy use efficiency was higher in flat bed CT treatment and this phosphorus foliar spray treatment. Lysine and tryptophan content in maize grain were significantly higher in this 50% P plus PSB plus MF and 2 foliar spray of DAP treatment. Ranjit PS, under the chairmanship of Dr. Dinesh Kumar, studied on phosphorus management in wheat aerobic rice cropping system. He had 3 phosphorus application method supplemented with 3 phosphorus doses, and it was found that integration of band placement of 30 kg P per hectare in wheat and aerobic rice was best for increasing the productivity, profitability, quality as well as resource use efficiency and energetics in these crops and cropping system. Found that agronomic efficiency as well as partial factor productivity of phosphorus in wheat and aerobic rice was higher in the band placement treatment as well as in 20 kg P application treatment. However, the phosphorus pools was significantly higher in this basal incorporated treatment and 40 kg P application treatment. Himanshu Shekhar Gowda, under the chairmanship of Dr. Y.B. Singh, studied the impact of enriched organic nutrient sources and establishment method on rice and their residual effect on zero till wheat, and he found that CT rice followed by zero till wheat gave higher system productivity as well as system net return than aerobic rice zero till wheat system, and enriched nitrogen sources gave higher values of system productivity as well as system net return than other treatments. He observed that City rice followed by zero-till wheat system had higher global warming potential than aerobic rice zero-till wheat system. Similarly, enriched nitrogen had higher global warming potential than RDA. And aerobic rice followed by zero-till wheat and enriched nitrogen led to higher system water use efficiency as well as system irrigation water productivity. There are 9 publications from student thesis, 6 are in greater than 6 NAS rating. Students participated in 7 international and 6 national conferences. They also had extracurricular activities. Susmita Tripathi participated in English debate competition, Hindustani vocal song competition, Monu SM 4x400 meter relay, walkathon debate competition. Our students also have received several awards and recognition. There are 3 students got Best Poster Award, and 1 student was Academic Council representative, and 1 student ambassador. Our students are well placed as Assistant Director of Agriculture Rajasthan, SMS KVK, Indian Institute of Horticulture Research, lecturer in Agriculture College Odisha, and a scientist. And most of the MSc students are pursuing PhD program. Students also received UGC JRF NET fellowship and CMIT CCAP fellowship, and division conducted 1 training program. Our faculties also received several awards and recognition like ICR Bharatratna Dr. C. Subramanian Award, Sriram Puraskar, Outstanding Scientist Award, Young Scientist Award NAS, and ICR Best Multidisciplinary Team Award, and several other awards. I am highly thankful and grateful to Director IRI, Dean Director Education, Joint Director Research, Joint Director Extension, scientists of Agronomy Collaborating Division, students, other staff of Agronomy and IRI for their help, cooperation and sincere advice. Finally, I thank one and all.
Thank you, Dr. Das. We now have the presentation from Environmental Sciences by Professor Surya Naresh Kumar.
Good afternoon, everyone. I welcome all you to this presentation on academic achievements of environment science students at the Center for Environment Science and Climate Resilient Agriculture. At the center, we have multidisciplinary and integrated faculty covering 7 disciplines, conducting research on environment and climate resilient agriculture. The mandate of the center is to conduct basic and strategic research for environment resilient sustainable agriculture, impact postgraduate education and training on agricultural environment and interrelationships, and to provide advisory services on environment and climate change in agriculture. The center has 18 scientists, 15 of them are faculty members. Center so far has produced 86 MSc students and 54 PhD students. Currently we have 20 MSc and 25 PhD students on roll. Our students are well placed in agriculture research services, state agriculture universities, as well as in central pollution and state pollution control boards, and also in several other organizations, including some of them working abroad. Coming to the significant research achievements of postgraduate studies under environmental sciences during 2020, that the research was conducted in 2 thematic areas, climate change and agriculture, in which 3 MSc and 1 PhD students have conducted their research, and another thematic area is environmental pollution, under which 2 MSc and 1 PhD students have conducted their research. Coming to the climate change and agriculture thematic area, one of our students, Mahesh Kumar Malav, has conducted research on greenhouse gas emissions in rice, wheat, mung bean cropping systems under organic and conventional practices. He also quantified the soil physicochemical parameters and soil microbial parameters to relate them to GHG emissions from organic and conventional agriculture practices. He has grown rice, wheat and mung bean with 7 organic sources of nutrients and also with the synthetic fertilizers. Results indicate that the cumulative methane and carbon dioxide emission from rice, wheat, mung bean system were higher from the plots receiving organic source of fertilizers, while the cumulative nitrous oxide emissions were significantly lower. The global warming potential among the treatments ranged from 500 to 1600 kg CO2 equivalent per hectare, while the organic treatments reduced the global warming potential to the tune of 35% in different crops. The results further indicate that the soil enzymes like beta and alpha glucosidase have positively correlated with the emission of methane and carbon dioxide. Therefore, the replacement of conventional management with organic practices could reduce the global warming potential and therefore needs to be considered for future development of sustainable farming systems. Another student, Mr. Aftab Kairadi, has conducted his research on effect of elevated temperature and carbon dioxide on the yield attributes, seed and grain quality of rice grown in 3 levels of nitrogen application. He conducted his research in temperature gradient tunnels with rice variety Pusa 44 with a combination of CO2, temperature and nitrogen. Findings indicate that the elevated CO2 reduced the harvest index of rice crop, whereas the seed quality was also reduced in the plants grown under elevated CO2 and temperature. The study indicates that the application of 25% nitrogen over the recommended dose significantly improved the growth yield as well as the grain quality and the seed quality of the rice crop. Another student, Mr. Savananda Darji, conducted his research under the guidance of Dr. Renu Singh on the topic transformation of nitrogen in wheat field under integrated nutrient management. He conducted his research with wheat variety HD 3086, the temperature regime 6, and also the fertilizer combinations with azotobacter and mycorrhiza and NPK. His research results indicate that the ammonium volatilization loss and denitrification losses were significantly higher in the plots receiving nitrogen, phosphorus and potassium through synthetic fertilizers, while the application of azotobacter and mycorrhiza significantly reduced these losses. Ms. Babetlan conducted her research on impact of plant growth promoting microorganisms on the nitrogen use efficiency and yield of corn in breadlines. She has conducted her experiments in phytotron conditions as well as in the field conditions to screen the germplasm lines for their response to PGPMs. Results indicate that the PML46 and PML36 have responded significantly in terms of yield and nitrogen use efficiency. Also, the partial factor productivity for nitrogen was improved, indicating that the application of PGPMs offer a sustainable approach to improve yield and nitrogen use efficiency of maize crop. Coming to the second thematic area, environmental pollution. Mr. Sandeep Kumar has conducted his research on heavy metal contamination in cultivated and marketed vegetables in Delhi region, and he also assessed the risk to human health. For this, he has collected samples from 5 cultivated sites and 2 regulated market sites. He analyzed the heavy metals in the collected samples in 6 vegetables as well as in the soil and water samples. He also calculated the risk assessment indices. The results indicate that the average concentration of lead and cadmium content were higher in soil samples, while in water samples, cadmium content in some of the places exceeded the permissible limits. In all vegetables, only cadmium content exceeded the World Health Organization permissible limits of 0.02 microgram per gram. Among the vegetables, spinach and okra had a higher transfer factor from soil to plant as far as the cadmium, chromium and lead are concerned. However, no hyperaccumulation was noted. The targeted hazard quotient of arsenic and mercury had higher values, indicating non-carcinogenic effects over long-term consumption of contaminated vegetables. The higher MPI and health risk index in spinach and okra indicate metal toxicity in specific samples and health risk with the continued consumption. Another student, Mr. Pankaj Kumar Patel, has conducted his research on screening of wheat varieties for air pollution tolerance. For this study, he has taken 10 wheat varieties and he also monitored the air quality during crop growth period. The air pollution tolerance index was also computed. The results indicate that the APTI has a high positive correlation with the relative water content and ascorbic acid. Among the varieties, HD2967 and HD2894 could tolerate air pollution and could give good yield. In another study, Mr. Ravi Teja has taken work on uptake and partitioning of titanium from titanium dioxide nanoparticles in rice. For this pot experiment on Pusa Basmati 1121, he has applied titanium through two different sources by two different methods of application in different doses of titanium dioxide. Results indicate that most of the titanium from nano titanium dioxide is fixed in recalcitrant pool of the soil. Nano titanium dioxide at the rate of 10 mg titanium per kg soil may be considered sensitive for soil enzyme activity as well as microbial population. Nano titanium dioxide imparted beneficial effect on rice plant growth up to. Coming to the other academic activities of the center, during the WIBO Summit, a session on climate change impacts and adaptation technologies was conducted on 8th October 2020 with myself as the convener. Apart from that, we have conducted Ozone Day special lecture by Dr. Madhulika Agarwal of Banaras Hindu University. We also have conducted the training program on simulation modeling and also several workshops and online meetings related to the project activities. One of our faculty, Dr. Nivendu Gupta, has visited UAE to participate in transforming food and agriculture to achieve sustainable developmental goals and global food security. During this year, we have inducted one faculty, Mr. Sandeep Kumar, who is going to get his PhD degree during this convocation. Under the study circle activities, we have conducted two special lectures by the experts on motivation at workplace and stress management in order to develop the soft skills of the students as well as faculty. I am very happy to mention that this year our students have done exceedingly well as far as the awards and extracurricular achievements is concerned. My special mention is to Ms. Pooja LR, who has been invited by the Government of India to witness the Republic Day parade as one of the meritorious student of the year. Special mention is also to Ms. Mamta, who bagged several awards in other activities, including academic achievements. This is the list of the extracurricular achievements by our students in different fora. This is a list of awards and fellowships to the students, and we have 15 students in PhD who are having UGCJRF, ICCR fellowship, IAFS fellowship or ICRJRF fellowship. Our faculty are well recognized during this year, who have been bestowed with several awards or responsibilities at national and international level. This year we have 9 research papers coming out of students' work, with 8 of them are having impact factor between 0.1 and 3, with highest impact factor of 2.14. Faculty have published 34 papers. 22 of them are having impact factor up to 4. Between 4 and 5, 6 papers, and 1 paper is having impact factor of 5.03. Thank you very much for your patient hearing. I thank my students, faculty, staff, skilled labor and farmers for their consistent support for doing all this work. And also, we at the Seskara family thank IARI authorities, PG School, Associate Dean, Dean, Joint Director Research and Director of IRI for their constant encouragement and support. Thank you one and all.
Thank you, Dr. Naresh. We now have the presentation by Dr. Radha Prasanna from Microbiology.
Experts, respected director, dean, joint director, heads of divisions, my professor colleagues, dear scientists and my dear students. I am here to present the significant educational achievements for the year 2020 from the Division of Microbiology. We have 19 faculty members, out of which 18 are research guides. We have a total number of 47 students on rolls now, out of which 16 are MSc and 31 are PhD students. Our faculty is extremely expertise in different areas of microbiology, and despite the COVID scenario, they have performed excellently not only in terms of teaching, but also research work and published in several peer-reviewed journals. The transitioning to the online mode of teaching. Here, I wish to place on record my praise for my faculty who rose to the occasion and performed excellently and took up the challenge despite the COVID scenario posing problems. They also were a major source of support for me for transitioning from the trimester to the semester mode of teaching, and particularly in the online mode. They not only helped in the preparation of the courses, but also in the implementing them successfully. Our faculty has won several awards, among which Dr. K. Annapurna was awarded the Fellowship of the Academy of Microbiological Sciences, while Dr. Ramakrishnan has been selected as a committee member of the Ministry of New and Renewable Energy, Government of India. We have a DBT TWAS postdoctoral scholar in our division, and one of teams of microbiology, Dr. Annapurna, Dr. Ramakrishnan, Dr. Archana Suman and Dr. Govindsamy visited US as a part of the IU SSTF program, in which a workshop was held in US. Our faculty has been involved actively in organizing and being invited speaker in various webinars, in various workshops, as well as participating in conferences as panelists and conducting training. Our students are not far behind. They have also been attending a large number of workshops and also webinars and improved their skill sets. And many of them have also presented posters in different conferences and also won awards. Some students have also participated in the Inter-Agri Sports Meet. And two of our students have also got employment in the state government services. During this convocation, 7 PhD and 4 MSc students are receiving degrees in the following theme areas. The first student, Ms. Aditi, whose chairperson is Dr. Anpurna, worked on identifying the different N level which is most optimal, as also when it should be applied for effective symbiotic association of bradyrhizobium with soybean genotypes. They selected 6 soybean genotypes and 6 root nodulating bacterial strains and set up a hydroponic experiment with different N levels and found that there was different changes in not only the root Cas712 plus Cas1 combination as the most promising, and addition was then evaluated both basally and addition after 15 days after inoculation. And they found that the higher doses at the time of nodule initiation was more inhibitory. And this was supported by the expression studies on the early nodulin gene. Mr. Dipankar studied the sulfur oxidizing bacteria from different econiche for their role in mobilization, and they isolated 127 sulfur oxidizing bacteria. His chairperson was Dr. Archana Suman. They then undertook PGP characterization, and they were able to identify 17 of them, which they characterized further, and they were found to belong to 3 genera, Thiobacillus, Bacillus, Klebsiella. And then a pot experiment was taken up with and without the addition of elemental sulfur to 3 types of soil, which indicated the significant role of sulfur oxidizing bacteria not only in the transformation of soil but also improving plant growth parameters and uptake of S by the plant. Mr. Marag, a PhD scholar, was working on the influence of cyanobacteria in rice crop. Here they tried to understand the rhizospheric diversity. They took 8 varieties of rice and they identified 85 cyanobacterial strains, 71 from the rhizosphere and 14 from the other plant parts, out of which 70 were heterocystous forms. Further, genera-wise distribution showed that Anabina and Nostoc contributed to 72% in the overall abundance. The diversity indices were also very different for the different places from where these samples were isolated. The interactive effect of N-fertilizer and cyanobacteria inoculation was studied both in pot experiments under natural and controlled conditions as well as in the phenomics facility. The salient findings were that 75% N and cyanobacteria inoculation exerted positive influence on selected plant attributes, particularly N assimilation enzymes, as well as root biomass and also grain quality. The phenomic study also showed that significant influence was exerted by cyanobacteria inoculation on plant traits. Thus, such cyanobacteria can help in saving about 20 to 25% nitrogenous fertilizers. Mr. Sai Prasad, under the guidance of Dr. Archana Suman, studied the seed-borne endophytes in diverse wheat ecotypes. Towards this, they developed a new growth medium, WMM, for the isolation, and a total of 220 isolates were there, among which 62 were selected based on the plant growth promoting traits and lytic enzyme activity. And these were further undertaken for analysis of in terms of their genotypes, and they were found to belong to 7 different genera with 20 distinct species. And the most promising among all these were identified as Pseudomonas putida, Pantoia agglomerans, and Bacillus cereus. The colonization of these endophytes was also undertaken using bright field microscopy and differential staining, as well as GFP tagging and confocal microscopy. This showed that these 3 strains were able to colonize the plant system and also bring about significant increase in grain yield in 3 wheat species using pot studies as well as field studies. The conclusion was that the seed endosphere is an important microbial habitat which can have immense potential for developing novel bioinoculants. Ms. Vijaya Rani, who worked under the guidance of Dr. Rajiv Kaushik, was able to isolate certain methane utilizing bacteria and develop a co-inoculation protocol for not only reducing the methane emission but also improving the plant growth. Towards this, they isolated samples. They isolated bacteria from different phyllosphere, rhizosphere and round rhizosphere of 5 rice growing regions of India. And among these, they isolated ones. They were found to group into 13 bacterial and 2 yeast genera. Out of the 15 genera, most number of isolates belong to Burkholderia, followed by Hypomicrobium. Based on kinetics of methane utilization, Methylobacterium oryzae showed the highest methane utilization, while Penibacillus polymixa was selected as the best under the PGP traits. So the co-inoculation of the strains were undertaken with through field experiments. The first one was using different N sources such as urea, DAP plus urea and FIM, wherein they were able to show a significant decrease in the cumulative methane emission. The second study was on different rice varieties. Here too they were able to show a significant reduction in the methane emission. But more interestingly, this also brought about significant increase in yield, quality of rice straw and grain seed protein, as well as biofortification in terms of iron and zinc. Such potential organisms can be exploited for developing novel biofertilizers for promoting the growth and yield of rice. Coming to the theme 2, microbe-mediated abiotic stress management, Mr. Anaya, under the guidance of Dr. Abraham, undertook the characterization of desiccation tolerance in a cyanobacterial isolate. They isolated several cyanobacteria from soil biological crust, and based on growth characteristics, they identified Anabaena species for further investigation. They were able to show that this strain was unique in producing very high levels of pigments, phycocyanin, and also showed a differential fatty acid profile when subjected to desiccation in terms of MUFA, PUFA, as well as saturated fatty acids. And also the 2D analysis of the Anabaena species subjected to desiccation showed 45 differentially expressed proteins, among which 28 were upregulated, whereas 17 were downregulated in response to desiccation. Further characterization is going on. Ms. Asha Devi, who worked under the guidance of Dr. Sangeeta, tried to understand the interactions between osmotolerant rhizobacteria and mustard under water deficit stress. Towards this, 11 osmotolerant bacterial cultures were taken and screened, among which 2 strains, Bacillus cereus NAD7 and Bacillus casamensis strain MK6 were selected. They exhibited superior performance in yield and growth. The attachment ability of both the strains was found to be affected due to osmotic stress. Reduction in Cu was recorded, but EPS of both the isolates recorded an increase and also recorded changes in terms of the qualitative and quantitative amounts of the different sugar monomers. Osmotic stress and inoculation both had positive influence on both the quality and quantity of phenolic compounds also, and a total of 29 phenolic compounds were identified by LCMS. Inoculation also brought about significant enhanced photosynthetic traits. Protein profiling showed that in both the strains there were different number of protein spots and there was a differential expression in response to osmotic stress. The phytohormone IAA content was significantly increased due to inoculation and positively correlated with the transcript expression of the phytohormone biosynthesis gene as shown by real-time PCR. Also, the expression of drought tolerance associated gene under drought stress was found to be significantly upregulated by the inoculation of these 2 strains. Overall, they concluded that the vegetative stage of the crop was more responsive to rhizobacterial inoculation than reproductive stage under drought. Second, it highlighted the potency of rhizobacteria in reducing the negative impact of drought stress in mustard. Ms. Niveta, PhD scholar, also worked under Dr. Sangeeta. They tried to understand the plant bacterial interactions, particularly in relation to seed germination and root colonization. They took up the 2 osmotolerant cultures, Bacillus MRD17 and Bacillus casamensis MKS6, and they were able to show that the inoculum density has a major influence on seed germination, and even high CFU can actually inhibit the seedling growth. Now, water deficit stress did not have any impact on both these populations on the roots, and while one of them is an endorhizosphere isolate and a rhizoplane colonizer, while MK6 was a rhizoplane colonizer. Now, coming to the rhizobacterial inoculation, it was found to have a major impact on the expression of genes involved in phytohormone synthesis. Also, osmolytes proline and glycine betaine accumulation was much reduced upon inoculation with these rhizobacteria. The profile of flavonoid sugars and phenolics showed immense changes. Now, the exudates of these plants were found to show changes particularly in the content of the organic acids, and acids such as glutamic acid and myristic acid were observed to be present only under stress. Osmotic stress was also having a positive influence on cell wall hydrolytic enzyme production. The expression profile of the rhizobacterial cell wall protein differed significantly due to influence of the root exudates. And presence of these exudates also changed the expression of nearly 50% of the proteins. So in the conclusion, they were able to show that these two strains improve seed germination, and if provided an optimum population density, they had excellent colonization ability and could be used for drought stress alleviation in mustard. Theme 3, Agri-biomass Degradation and Value Addition. Ms. Shreya Virmani, an MSc student under Ms. Anju, worked on the microbial conversion of lignin and profiling of its degradation products. They selected 5 different fungal strains and 6 bacterial strains from leaf litter. All these strains were found to produce lignin peroxidase, while only 2 fungal strains were found to produce laccases. The 2 most important bacterial strains, the potent ones, were identified as S1 and S2 as Acinetobacter baumannii, and the fungal strains were identified as Aspergillus niveolens and Aspergillus niger. This S2 strain also showed the highest lignin degradation of 46.6%, while among the fungal strains, Aspergillus niger showed 76.8% lignin degradation. Solid state fermentation led to the value-added compounds such as vanillin and coumarin, which were produced more by Aspergillus niger, while coumarin, cyanopic acid, and coniferyl alcohol methyl catechol was produced more by Acinetobacter baumannii S2. Now, both these strains can be potential organisms for obtaining value-added products using agro-waste. Coming to the PhD program of Ms. Shruti, who worked under the guidance of Dr. Livleen Shukla, they were trying to identify strains which have lignolytic properties, because this could help to degrade the rice stubbles, which are a major problem nowadays because of the burning issue. Now, they were able to identify 19 morphologically distinct fungal strains, and among them 3 strains were found to have promising activity. These were identified as Aspergillus terreus, Aspergillus fumigatus, and Alternaria arborescens. Now, the highest activity of these enzymes was found when rice stubble was chopped to 2 centimeter length, and this also reduced the CN ratio considerably. And these 3 isolates were compatible with each other, and their consortium, when applied at 15% weight by volume by weight, was found to be the best because it brought about significant changes in the composition, as you can see by the FTIR profile. HPLC analysis revealed that lignin intermediates such as vanillin, vanillic acid, and syringic acid, and these activity were high when rice stubbles were amended with nutrient NPK at 70% moisture. A formulation of 3% jaggery medium amended with 1.5% PVP was found to maintain shelf life of the consortium up to 12 months. In field, the highest degradation potential was observed when both the lignolytic consortium and Pusa decomposer were applied together on rice stubbles chopped with mechanical chopper. This consortium can be further optimized for effective bio-based management. Mr. Deepak Kohli, under the guidance of Dr. Sunil Pabi, tried to develop spirulina-enriched food and evaluated their nutritional quality. As you know, spirulina is considered one of the superfoods, and during their preliminary screening, they were able to identify 2 spirulina strains, which they further optimized the physicochemical parameters as well as media components for high biomass nutraceutical production, as well as for enriched food preparation. RNSEM studies were also undertaken and validated with the predicted concentrations of the nutrients. Spirulina incorporation was found to lead to increase in proteins, lipids, total phenols, flavonoids, and other important compounds without causing any detrimental changes to the textural, cooking, and sensory attributes. Among the different foods prepared, the pasta enriched with 12.5% dried and 20% fresh spirulina biomass was rated most acceptable by the sensory evaluation. These are some of the foods which were prepared using spirulina, which was cultivated in raceway ponds in a semi-controlled greenhouse, and the powder as well as the biomass was used for enriching the pasta, as well as cookies were prepared, as well as the granola bars were prepared, as you can see here. To end, I would like to thank the authorities, the director, dean, and Dr. Manjia, and the staff of the post, head of the division, faculty, colleagues, advisory committee members, experts who evaluated the thesis, the facilities, the phenomics, the NPF, as well as the technical, administrative, and supporting staff members of both Division of Microbiology and CCUBGA. Thank you very much. I wish to end with the quote that life is beautiful when you know what to ignore. Thank you very much.
Thank you. Thank you, Dr. Radha. Thank you. Now we have the presentation from Soil Science and Agricultural Chemistry, Professor S.P. Datpa.
At the outset, I welcome you all. It is indeed my proud privilege to present some of the most important educational achievements for the discipline of soil science and agricultural chemistry. This year, in all, 9 MSc and 6 PhD students are going to get their degree. Key research areas include enrichment, mineralization, stability of soil organic carbon, enhancing nutrient use efficiency, development of rapid techniques for soil fertility assessment, in-depth study on transformation of nitrogen and phosphorus in soil, use of non-conventional sources of plant nutrients, alleviation of micronutrient deficiency. Final one is use of biochar and co-compost for reclamation of acid soil. Now coming to the first theme, that is soil organic carbon. Soil organic carbon has been considered a soul of soil, which is of utmost importance for sustenance of soil health, nutrient supply capacity, as well as environmental quality. Hence, Rajkumar Meena, under the chairmanship of Dr. Mahesh Chand Meena, assessed the soil organic carbon and nutrient fractions under different land uses, where the highest level of total and level fractions of carbon were recorded under native forest, followed by horticultural and agricultural land uses, which is also reflected in nutrient supply capacity of soil. In our climatic condition, build-up of carbon is important, but stability of carbon is more important. Hence, Saurabh Das, under the chairmanship of Dr. Ranjan Bhattacharya, worked out the decay constant, Q10 values and activation energy of carbon mineralization using Stanford and Smith model as well as Arrhenius equation. Results indicated that there is considerable variation in these parameters under different land uses, and microaggregate carbon was more temperature sensitive than macroaggregate carbon. In the similar line, Rishabh Kumar Didavat, under the chairmanship of Dr. V.K. Sharma, assessed the impact of long-term organic farming practices on soil organic carbon as well as nutrient pools. Results indicated that both total as well as oxidizable pool of carbon were increased with the increasing rates of manure application, which is also reflected in nutrient supplying capacity as well as crop productivity. So this is all about organic carbon in conventional agriculture, where the substantial build-up of organic carbon is difficult proposition. Now coming to the conservation agriculture, Amit Das, under the chairmanship of Dr. V.S. Dwivedi, assessed the stability of soil organic carbon, where substantial increase in soil organic carbon both in macro and micro aggregates were recorded due to residue retention. Decay constant and Q10 values of carbon mineralization were decreased with increase in residue retention, particularly in zero-till plot. Their studies also indicate that carbon in CA is physically protected, especially in macro aggregates, and more importantly, this pool of carbon is relatively temperature insensitive. Coming to the second theme, that is development of novel fertilizer products. In India, annual fertilizer nitrogen consumption is about 18 million tons, and out of which 11 to 12 million tons are lost to the groundwater, water bodies and atmosphere due to its low use efficiency. Hence, Ashish Kumar, under the chairmanship of Dr. K. M. Manjaya, synthesized and evaluated nanoclay polymer and nanoclay biopolymer based slow release nitrogenous fertilizers, where results indicated that using these novel products one can curtail the rate of nitrogen application by about 25%. Use efficiency of phosphorus applied through conventional fertilizer is less than half of that of nitrogen due to high affinity of phosphorus for different soil constituents. Abhijit Sarkar, under the chairmanship of Dr. D.R. Biswas, synthesized clay polymeric and natural oil-coated DAP, and it is evident that release of nitrogen and phosphorus from this new formulation is slower than that that of DAP. Sustained and prolonged release of phosphorus is also evident in soil as compared to that of DAP. Uptake of nitrogen and phosphorus by wheat from soil treated with these two new products was higher than that of DAP. Clay polymeric coating is biodegradable, where half-life ranges from 23 to about 70 days. Coming to the third theme, that is development of rapid soil testing method. As the conventional soil testing method is tedious, cumbersome and time consuming, Ali Rafat Ali, under the chairmanship of Dr. Nayan Ahmed and in collaboration with Agriculture Physics Division with Dr. R.N. Shahu, attempted to assess the soil fertility using airborne imaging spectrophotometer. His study area was confined to Karnataka and Andhra Pradesh. Soil samples were collected and analyzed for important soil fertility parameters. Reflection data of the sites were acquired using airborne Aviris NG sensor. At the same time, spectral data in the laboratory were also collected using ASD, and soil spectral library were generated. Based on this data, multivariate regression models were developed using various approaches. Results indicated that models based on artificial neural network is most effective in predicting various soil parameters based on spectral data. Since this technique is empirical in nature, more case studies are needed to be conducted. Scientific basis of relationship of soil and spectral parameters also need to be established. Coming to the fourth theme, that is transformation of nutrients in soil. In fact, the knowledge on dynamics and transformation of nutrients help us to predict and manage the availability of nutrient in soil. Surya Prakash, under the chairmanship of Dr. Sarvindra Kumar, studied the distribution of nitrogen in different fractions as well as microbial functions in cotton wheat system under conservation agriculture. Conservation agricultural practices like zero tillage and residue retention led to build-up of nitrogen more in hydrolyzable pool as compared to conventional tillage. What does it imply? It implies that immediate and long-term supply of nitrogen are enhanced due to CA practices. In the similar line, Manoj Burman, under the supervision of Dr. Mandira Burman, assessed the effect of applied silica on dynamics of phosphorus in acid soils because the activity of phosphate and silicate ions in the soil solution is governed by the common components in soil. Results indicated that sorption of phosphorus is decreased in presence of silica while desorption of phosphorus is decreased, leading to the increase in availability of phosphorus, which is reflected well in P uptake data by wheat. Sayan Mukherjee, under the chairmanship of Dr. D.R. Biswas, studied the phosphorus transformation vis-a-vis phosphorus supplying capacity under low input organically managed system. In fact, organic treatments enhance the labile fraction of P at the cost of non-labile residual pools, which is also evident in the Bray P and which is available P. Application of organics and lime also modified the solid solution equilibrium of phosphorus, leading to the reduction in its fixation capacity. Coming to the fifth theme, that is use of non-conventional sources of plant nutrients, this area of research is important, particularly in the face of escalating price of chemical fertilizers and also our complete dependence on the import of potassic fertilizers. In this regard, Siddhartha Shankar Biswas, under the chairmanship of Dr. D.R. Biswas, studied the efficacy of low-grade rock phosphate and waste mica in enhancing the phosphorus and potassium supplying capacity of soil. Results indicated that The solubility of this material could be enhanced by treating with phosphate and potash solubilizing microorganism as well as organic acids. From the uptake data, it is also clear that these products are capable of substituting chemical fertilizer considerably. In our country, a huge amount of sludge is being generated, and this is good source of organic carbon as well as major and micronutrients for plant. But the use of this sewage sludge is mainly restricted due to the presence of toxic trace elements in it. In this regard, Mahipal Chaudhary undertook detailed study which indicates that there is wide variation in metal content among the sources and seasons of sludge generation. He also worked out translocation pattern of metals in different plant parts of rice, wheat, soybean, and mustard. Further, he has gone for risk assessment in terms of hazard quotient for intake of metals through consumption of food material by human at the various level of sludge application. In the similar lines, Anil Verma, under the chairmanship of Dr. Mahesh Meena, assessed the dynamics of trace elements in an ongoing field experiment which was initiated in 2014. Treatments include the substitution and supplementation of nitrogen to the extent of 25, 50, 100, 200 and 300% of recommended dose of nitrogen. Results indicated that both quantity as well as intensity of trace toxic elements in soil was increased with increasing rate of sludge, and these metals are mainly accumulated in oxide and organically bound fractions. Further, yield data as well as values of hazard quotient indicated that fertilizer nitrogen can be substituted to the extent of 25% with sludge. Coming to the sixth theme, that is alleviation of iron deficiency. In fact, this is an important area as deficiency of iron is one of the major constraints, particularly in upland conditions. Hence, Kiran Kayar, under the supervision of Dr. R.N. Pandey, screened 50 soybean genotypes based on their susceptibility and tolerance to iron deficiency. And experimental results indicated that performance of iron efficient genotypes was better under both ambient and elevated condition of carbon dioxide and temperature as compared to inefficient one. Further, integrated use of iron efficient genotypes along with ferrous sulfate and herbuscular mycorrhiza was effective in combating the deficiency of iron. Over the years, biochar have mainly been used for enhancing carbon sequestration in soil to improve environmental quality. Co-composting of biochar is relatively a new concept which have been used to improve the soil condition. Pooja Nayan, under the supervision of Dr. T.J. Purkayastha, prepared a series of biochar and biochar co-compost and evaluated their efficacy for reclamation of acid soils. Results indicated that biochar and biochar compost prepared using gram residues are more effective in raising the pH of acid soil. There are 21 scientists in our division, and we could publish as many as 71 research papers, and I am very happy to present that out of the 71 papers, 6 papers had NAS rating of 10 and above, and another 8 papers come in the range of 8 to 10. Coming to the honors and awards received by our student, Mr. Kingshuk Modak received Best Master Research Presentation Award 2020. Ms. Kavita Pijatov received Best Article Award. Mr. Arko Prabha Roy received Best Student of the Year Award from ICAR IARI. He also received Best Master Research Presentation Award 2020. Mr. Anit Das received second prize in oral presentation, and Mr. Khurshid Alam received third prize in poster presentation, both in international conferences. Dr. Devrup Das, he received Mosaic Company Foundation Award, and this is a prestigious award. Dr. Govinath also received Jagannath Raina Memorial Award. Coming to the employment details of the students, during the year 2020, 3 of our students, Mr. Rahul Mishra, Khushbu Rani and Avinash Das, they joined Agriculture Research Service. Another 2 students, Ansuman Das and Kingshuk Modak, joined Indian Council of Forestry Research and Education as scientists. Coming to the honors and awards received by our faculty members, Dr. D.R. Biswas, Principal Scientist, he has been elected as a Fellow in National Academy of Agricultural Sciences. Dr. Ranjan Bhattacharya, he joined editorial board as a member of 2 prestigious journals, Geodharma and Agriculture Ecosystem and Environment. Dr. Mahesh Chand Meena, Senior Scientist, he received Young Scientist Award from Mosaic Company Foundation. This is again a prestigious award. Dr. Abir De, he received the Scientist of the Year Award by NRM Foundation, New Delhi. Dr. Devashish Galui, selected as Indian delegate in the section of Agriculture Food Processing for Young Scientists, Conclave of Shanghai Cooperation Organization. This time, under COVID pandemic, we could conduct only 1 training on capacity building for generation of soil health cards. And finally, I am grateful to Honorable Director, Joint Director, Research, Education and Extension, Associate Dean, our own Head of the Division for adequately and aptly supporting the academic activities of the discipline. I also gracefully acknowledge the contribution of the faculty and students of the division. That's all. Thank you very much. Thank you.
Thank you. Thank you, Dr. Datta. So now we have come to the last presentation of this session, which is on water science and technology by Professor Man Singh.
Chairman of the session, Director IRI, Dean, faculty and students, my greetings to one and all. Mr. Chairman, sir, with your permission, I would just like to recapitulate that the life of the plant is much more directly dependent on water than is life in the animal kingdom. In contrast with animals, land plants live permanently in one place so that they need water right there. This knowledge prompted for irrigation-based civilization. There are several technological challenges like supply, delivery, distribution, reuse, and storage of surplus water. And thus, faculty and students of water science and technology are striving for that. We have a modest faculty of 10 with 30 students. This year, 2 students, Gokul Raj and Ankit Dahiya, will graduate and receive master's degree, and I would present their work right now. Gokul Raj worked on absorbent, super absorbent polymer and the early season cauliflower. He used the variety Pusa Suni, early maturing, and control treatment, and there were 4 gel application treatment. And we can see the picture of the field that rain-fed plot cauliflower, it is very obvious how they are growing. And the result, if you look at, if we see the irrigation regime or the irrigated plots, there is a just 20% increase if we compare with the irrigated one. So that means that 20% is the contribution of gel application. Whereas if you look at the rain-fed and this, there is 200% increase. And if you look at among the gel application average with rain-fed 80%. This means the contribution of gel application is quite big in rain-fed areas. So the message is that superabsorbent may be used for production of early maturity variety where monsoon withdrawal is early. Now the Ankit also worked, he studied for impact of wastewater irrigation on marketable yield in chrysanthemum crop, and also he wanted to assess the effect of wastewater irrigation. Now he used two water, groundwater as well as wastewater, and he chose the scheduling criteria 25, 50 and 75% maximum allowable depletion of moisture. And we see the wastewater plants used by wastewater, the plants are very healthy and the flower numbers are also quite high. You can see that the water use efficiency and yield is something like this. You see the T4, T5, T6, they are wastewater treated ones and they are having very higher water use efficiency as compared to the groundwater. And here the results indicate that the vessel life of the flower is also higher in case of wastewater treated irrigation. And quality and quantity of crop. This year the journal publication is 24, which if compared to last year, about 26% increase. And faculty recognition and award, for me, I felt good when I was invited by ICR to speak on Gandhian philosophy during 150th birth anniversary of Mahatma Gandhi. There are some other committee. I'm expert member on some committees of Ministry of Jal Shakti. Dr. Kaur also is a well-recognized scientist. This year, she got award for most inspirational educational leader, outstanding scientist, and so on. Dr. Manoj Zander is a Secretary General serving for ISAE. He was invited for Agri-biomass India keynote speaker. Sushma Siddhasri, outstanding scientist award, and things like that. Similarly, some young faculty also, they have been recognized for their work. Students' participation was not much because most of the students were off campus. The 2020 reason well known to all of us. Now, this was the collective effort of the group of faculty members and the scientists of water science and technology. They demonstrated a live watershed management model. This was witnessed by all important people in the country who have a linkage for water and agriculture. So this is what was a successful demonstration. Now this year, Jalopachar is successful technology, which was awarded in December 2017. Dr. Ravinder Kaur led that group, and a success story for that was published and released in the 25th regional institutions and others like Sikopur, Goa, Bangalore, Jovner, and many more places of that sort. And that year, under ICR Swachhta Action Plan, she spent 38.43 lakhs rupees. This is if we imagine always the students are diligent, then the diligent students bolster water science and technology. The faculty is the strength for guidance and teaching and training to the young students. And finally, before I conclude my talk, I would like to read this message just to remind all of us, take up brain, muscles, nerves, every part of your body be full of that idea and just leave every other idea alone. This is the way to success. That's what Swami Vivekananda pronounced. And I feel that all of us, if you could follow this, we will certainly succeed. Thank you very much. I acknowledge to the faculty members and students, the Dean, Associate Dean and staff, Director, IRI and others for their cooperation and support. Thank you so much.
Thank you. Thank you, Dr. Singh. Thank you. So we have completed all the 7 presentations, all good presentations. So now I would like to request our chairperson, Dr. A.K. Singh, to kindly give his remarks on all these 7 presentations.
Thank you, Dr. Sima Jaggi. Well, hearing the work of almost 70 students and trying to, digest is a big task. So at the outset, I would like to congratulate all the students and the faculty for the various awards. Almost each division had some remarkable achievements on that field, and particularly for the publications, the quality of publications, because generally NRM was always stated to be having, less publication in the highly rated journals. So it's a very good sign that work is being conducted of that standard and being recognized outside. So congrats to all of them. As I mentioned, this natural resource management discipline is going to contribute a lot in the overall productivity and productivity enhancement and sustainable production. What I would like to just mention a couple of things which I have observed. One is that, of course, we have the inputs the NRM inputs are in terms of energy, in terms of fertilizers, in terms of water, and of course, agrochemicals, part of it is covered in the plant protection. So these are the inputs which are required, and we also know that the efficiency of these inputs is fairly low, and we have to improve the efficiency of all these parameters. And it is good to find that work is being done on that. I would like to just give a few suggestions then when, of course, in some of these presentations they have mentioned, but I would like to, particularly the work of PhD students, for MSc students, they are just working, is how it is being translated in the terms of enhancement of efficiency, increase, whether it is energy, whether it is water, whether it is fertilizers. Some of them have mentioned it, so it's a general statement, because ultimately the efficiency terms are going to matter. Second thing is that the PhD work that is being done, particularly if you look some of the disciplines like agronomy or soil science, how are they planning to transform or translate those results into recommendations which the farmers can adopt? That's very important, because a lot of studies are being done on efficiency. There are a lot of studies in which they have also indicated the benefit cost ratio or the increase in net returns. How do you take those practices to the farmers? I think that is very important, particularly to the work of PhD students. And also, again, and overall, I would like to mention that one has to always include 3-4 things when it comes to NRM. Of course, one is the reduction in the greenhouse gas emission intensity, which I think we are all looking forward. The other is the global warming potential. In some of these studies, it is mentioned, definitely. But I think when you talk of efficiency, it has to be reflected somewhere in that. Carbon sequestration has been talked about, because these are the results which are needed at the national level, and scaling up of these results so that we can stick to the commitments, fulfill the commitments made by the government for 2030 targets. And one more thing that I would like to see, at least from the PhD work, is what are the contributions of whatever result they have towards the ecosystem services, because natural resource plays a very critical role in the ecosystem services, which are not being really recognized. We do recognize in terms that we talk about lot about ecosystem services, but the valuation part, how do you give the benefit of that either to the farmer or to to the agency which is taking benefit of that? Somewhere it should be reflected in some cases. which I feel particularly, because ultimately these ecosystem services, some of them are, indirect, intangible, but they contribute a lot in sustainable production systems. So I think this is one aspect which can be looked out by the concerned departments. Now, coming specifically, I think we know that engineering division is doing a lot as far as coming up with these machines are being there. Of course, as I mentioned, their contribution in increasing the energy efficiency, reducing the greenhouse gas emissions, because all those activities are related to greenhouse gas emissions. And then how do you plan to convince the stakeholders, because we want mechanization to increase, but if you go to the field and look at the farmers, the level of mechanization has not gone much, except for very, advanced states like you can say Haryana, Punjab, but most states you still find mechanization is not being adopted to that level where it should be. They also worked on liquid fertilizers, but I think that was more with the protected cultivation. But I would like to see, and I think both soil science, agronomy, and engineering should see what is the potential of liquid fertilizers in the normal, non-protected cultivation, the routine cultivation. Is there some scope? Because a lot of work is being done outside on liquid fertilizer application in normal cultivation. So I think it's very important if somebody can have an idea of what is. One of the studies in that engineering was on canal water management, but it was a very routine report which they gave. They should have also given how you could match the demand and supply, because they were very obvious that the supply in the initial stages is more than the demand, and later on the demand is high, supply is limited. They could have given what should be the way about so that they can take care. One good thing I observed was for in many disciplines is that use of modern technology, whether it's remote sensing, machinery, then also I think now the government of India has okayed, use of drones. Guidelines are there. So I think probably an institute level project should be there on how, maybe it's already there, because I know some meetings were held on that, how to make the best use of this technology, because our farm holdings are very small. Drones can do a much better job than the remote sensing. And then the processing time of remote sensing images is there. So by the time it's very difficult sometimes to have a real-time decision making possible through the routine remote sensing. So, and of course, the use of modern data mining techniques is very encouraging, and I hope more and more of these techniques are used by other disciplines also, beyond the ANN technique. So one thing I would like to just mention in case of agronomy, they mentioned about radiation use efficiency enhancement through agronomic management. Now, radiation use efficiency is actually a characteristic of the plant. How could the agronomic management increase the radiation use efficiency? It was not clear. Maybe there was no time or limited time, they could not explain. But probably this need to use a proper terminology, because it's a property which is not influenced routinely by agronomic management. Totally. There are a lot of studies on intercropping, which was reported. Now, one thing is whether intercropping, we all know it has been, we have been reading and studying for quite some time, but how much is being really adopted by a farmer in the present setup, where everything is machine based and you have very, very limited machines in which you can do the operations on intercrops. So they have to see at the relevance of that. It's good to find that there are many divisions working on various aspects of conservation agriculture. My suggestion would be that they should have one data set prepared so that they can fill each division can fill some component, and ultimately they have to come up with a model so that they can predict what is going to happen when you do conservation agriculture for 5 years, 10 years, 15 years, 20 years, so that you don't have to really experiment every time with so many people. IRE has the advantage that you have experts in so many components. That will be the of course, I think we have been talking a lot about the role of microorganisms, and it's very good to see a lot of studies being conducted there. They are going to play a very important role, whether it is biofortification by increasing the efficiency of nutrients release, whatever it is, including the helping the plant, tolerate abiotic and biotic stresses. And of course, the crop residue is one issue which was quite highlighted, and it's going to be very, very important. In the fertilizers, I would say that there are some studies which are working on the fertilizers, polymer coated fertilizers, which have a release pattern not identical but similar to what the plant's requirement is there. So they are, I think somebody should be working on these. Maybe somebody is working because this was only certain of students, but that's very important so that the release pattern of the particular nutrient can match the plant supply. Some work should be done on these. Otherwise, a lot of students are doing. I know the phosphorus is important, potassium is important, and only very recently a study came that the phosphorus stocks in the world may not last for very long. So one has to look at rock phosphate, and some good studies are being done there. Again, there were some studies both in the environmental science and in water also about the use of wastewater, sludge in soil science. I think one of the things particularly where in WTC where they have presented for use of wastewater in flower cultivation. It's very important wherever you are using either sludge or wastewater, where will those toxic elements, heavy elements end up? Flowers you may not be consuming, but that flowers are being put in the house or disposed later on, once they dry up or they wilt. Similarly, whatever products are there, ultimately, where does that particular hazardous element end up and how to take care of? That's very, very important, because it should not end that you have grown flowers, they are a better bunch of flowers because of the nutrient composition. Ultimately, so the complete cycle has to be somehow taken care of when you come up, when you're using material like that. One more thing I would like to say, now many of these experiments, they have water and nitrogen or water and fertilizer interactions. But to have production functions, water and nutrient production functions, they should be having data of each treatment water-wise and fertilizer-wise. Normally, they give all the water treatments in one place, all the fertilizer treatments. So in that case, it's not possible. But if they have, they must be having, at least they should be having, for reporting, okay, it's all right that you highlight the impact of one particular treatment, whether it's irrigation or fertilizer, but they should aim that they can develop water nutrient production functions so that each treatment, whether it's water or fertilizer, we have the quantity of both. And then it's easy to work out an optimum combination. Otherwise, you have only selected treatments and you can only have a combination of either of them. Out of 4 treatments you pick up, this is the best one. Out of the other nitrogen you had, which is the best one. But the optimum could be somewhere in between 2 limits. So if you have those functions, basically some estimation is required. I think if they can probably include that as part of it, it will be. And it was also good to see about in one of the studies about the spirulina. I think nutrition is going to be very, very important. Iron deficiency is also studied by soil science. I think we have to look more at the micronutrients because these are the ones which are going to be important for the nutrition security. So I think these are some of the things, just some observations, random thoughts, which came to me during these presentations. Thank you once again for giving me this opportunity, particularly Dr. A.K. Singh, Director, IRI, and Dr. Agarwal, Dean, and Dr. Jaggi, you. And of course, once again, I would like to congratulate all the students and the faculty for the awards and the students for the degrees. Thank you very much. Over to you.
Thank you so much, sir. Director, sirs.
It's fine, Dr.
Yes, sir.
Rasmi.
Yes, sir.
I have no comments to offer. Excellent presentation and very well summarized by our resource person, Dr. A.K. Singh, including the aspects which generally we will ignore on the flowers when you keep in your drawing room or when you dispose it off, if it has heavy metal, how it is going to impact the whole cycle actually that he mentioned. So very valuable comments and on all aspects. So, and excellent presentation from all the professors, and they reflects the quality of work. And, my congratulations to each one of you, the professors who made the presentation. Thank you, and
Yes.
especially thank Dr. A.K. Singh for sparing, almost 2 hours time to be with us to see all these presentations and give his valuable suggestions and inputs. Thank you.
Yes, Dr. Seema, now we will wind up this session.
Yes. Thank you. Thank you so much, sir. Special thanks to Dr. A.K. Singh, sir, for chairing this session. And also thank you so much for your concluding remarks, your encouraging and inspiring words, and also